Hello, everybody. Good morning. I would like to say hello to everyone, to all of the participants to this event. I am Dr. Mohammad Khami, the moderator of this event from TUMS side. I'm so happy to be a part of this prestigious webinar and prestigious meeting between the Tehran University of Medical Sciences and Thammasat University. I'm sure that we will have a very fruitful meeting with the speakers. We have distinguished speakers from both sides. And okay, we can now, I think it's the time to start. If you agree, Dr. Baharat, hello.
Hello, Dr. Mohammad Khami. How are you?
Thank you very much. So I think we can start now.
Yes.
Okay, and then our first speaker is Professor Mohammad Hossein Nekufar. He is the Director of International Relations of Tehran University of Medical Sciences. He's an endodontist. And okay, Dr. Nekufar, the floor is yours.
Hello, everybody. Good morning, and thank you very much from both sides. And I'm very happy to be in the part of this very important event. And thanks to Professor Inglam and his colleagues that they help us to have this mutual work. What am I going to say in 5 minutes is about the importance of international collaboration. One of the very important things in these days in academic level is the international collaboration. The reason is because if we do whatever we are doing at the moment in our house and in our universities, we cannot understand what happened outside of our houses. So we need actually to start growing and start finding different links in different universities around the world and start making a very, very good network and transfer our knowledge to each other. And obviously our young fellows, our students can find a lot of new things and a lot of new area of research. And this research can help the next generation to use that and apply that for our patients, because the most important things for our universities, especially when the university is going to give service to the community, is to think about who is going to benefit from this result. The result of the research must be applicable. And the good things about this joint online program on oral cancer is the importance of giving service to the patient and to the community. And based on this very, very important international collaboration that we start now doing that, even when we look at the mileage and look at the distance between our countries, we may think that it is very difficult to arrange such things. But based on the virtual communication, based on this amazing platform that we are using these days, now the whole world is very, very small. It seems we are living in the same place with each other and we are working in the same place with each other. I remember that around 5 years ago, the Ministry of Health of Thailand came to Iran. And I was actually honored to meet the Excellency, the Minister of the Health of Thailand. And we understand that in Thailand, you have a very important network and very important public health service. So the importance now for us also is to learn from your side and transfer our experience to your side at the same time. So thank you very much for accepting to work with each other. And I'm very happy that we have a lot of participants from around the world, not just from 2 countries. This is also another important part of this event. And I really appreciate all of the people that helped for arranging this, and my office, and as a director of international relations of Tehran University of Medical Sciences, is always at the service to help and facilitate such communication and such collaboration. Thank you very much indeed, and thank you very much, Professor Ingram. Dr. Khami, I'm done. Dr. Khami, you are mute.
Thank you, Dr. Nefokar.
Sorry.
The next speaker I would like to introduce, he's the Dean of Faculty of Dentistry. He's also the Associate Professor in Implantology. Please welcome Dr. Inglam.
Thank you. Dear delegates and participants, I'm Samler Inglam, Dean of Faculty of Dentistry, Thammasat University. It is my great honor and pleasure to welcome you to online webinar on the topic of the multidisciplinary approach for management of oral cancer, organized by the School of Dentistry, Tehran University of Medical Science, and Faculty of Dentistry, Thammasat University. On behalf of the entire faculty, I would like to express our gratitude for this opportunity to be the co-host. The webinar will initiate the collaboration between our dental school, present information, exchange of the research experience in oral cancer. I wish to bring to our attendees a wide variety of speakers from the both dental schools in the multidisciplinary topics of oral cancer. I believe that this very first joint webinar between the School of Dentistry, Tehran University of Medical Science, and Faculty of Dentistry, Thammasat University will provide updates and insights about oral cancer for the audience. Finally, I would like to wish our guests, colleagues, speakers, presenters, faculty staff members, and students attending the webinar a prosperous future and great success. Once again, we are grateful and honored to have the opportunity to be the co-host. I look forward to seeing you on at the programs. Thank you.
Thank you very much.
Thank you very much, Professor Inglam. So I think it's the time now for Dr. Behara to introduce the first presenter from Thammasat University.
Thank you, Dr. Khami. Thank you, Dr. Nefokar and Professor Inglam for your opening remarks. It is my pleasure to you to introduce our first speaker for this session. He completed his BSc in biology with first class in Kasisat University, and also completed his master's in biological science with merit at University of Manchester, followed by PhD in cell and molecular biology at University of Dundee in UK. He's currently a lecturer in oral biology. His fields of interest includes genetic of cancer, cell and molecular biology of cancer cell. So it's my pleasure to introduce the first speaker, Dr. Ekharat Hitakomote.
Thank you very much, Dr. Bharat, for the introduction. So I hope you can see my slide now, right?
Yes, we can see.
Okay. Okay, thank you very much, and have a good morning for all of you in Tehran University Medical School Medical Sciences as well. I believe that is quite early there. So in my topic, I'm going to spend with you 30 minutes on this topic of the recent biomolecular aspects in oral cancer. So as you can see from the topic, it's going to be quite a complex story to understand in 30 minutes, but I try my best to make everyone understand. So my name is Dr. Ekharat Hitakomote, and I'm lecturer in the Faculty of Dentistry, Thammasat University. As my background, I'm not a dentist, so my knowledge is basically on cell and molecular biology related to oral cancer. I would like to start my first slide with this picture of genetics. So I believe because this genetics is the code of life and is central to our understanding in pathology of the diseases, including oral cancer as well. As you all know that drugs nowadays develop as the molecularly targeted drugs for the very specific and highly efficient therapy and have a lot less side effects comparing to the conventional therapy that we have in the past. So I believe that working together with the scientists, together with the clinician, we can achieve the goal for better treatment outcome for the patients in the future. We're talking about oral cancer. I would like to raise some problems that we can still have globally with the oral cancer disease. With the advanced cancer stage, with lymph node metastasis, which currently still a major problem. At the cellular level, there are high variation of genetic instability, or molecularly heterogeneity and complexity of subcellular abnormalities through oral carcinogenesis, which I have some of my data to show you related with this stuff. 50% of the patients with advanced oral cancer do not respond to therapy and develop metastasis, which is a major cause of death. And nowadays, limited curable therapeutic treatment is available for the patients. One successful story from the research is the drug called cetuximab, which is considered as the epidermal growth factor receptor inhibitor. Itself is the monoclonal antibody against this receptor because we now know that we have knowledge that is highly expressed in oral cancer patients as well. So better understanding.
I'm sorry to interrupt, Dr. Ekarat.
Yes.
But I would just like to say that you are, could you please switch to a presenter view?
Oh, you don't see my slides?
You see your slide,
Okay.
but it would be nice if you could present in the presenter view.
All right, I'm gonna change it a bit. Okay. Thank you.
Welcome.
Is it better now?
Yes, perfect. Thank you.
Is it okay now?
Yes, please.
Okay. All right, I'm gonna start all over again with this one. I'm talking about genetics because I believe that genetics is central for our understanding of the pathology of the diseases, including oral cancer as well. So nowadays we develop a lot of drugs that is based on the research. It is considered as a molecularly targeted drugs. For example, cetuximab, as I mentioned earlier. So still have a problem with this oral cancer because at cellular level, we know that there are high variation of genetic instability or molecular heterogeneity and complexities at a subcellular level through oral carcinogenesis. I have some of my data to show you related with this. And there are a lot of 50% of patients still have an advanced oral cancer, do not respond to standard therapy and develop metastasis. So I already mentioned that cetuximab is one of the successful story of the research has been done and is developed the drug against the EGFR receptor, which called cetuximab. So from what we understand, so better understanding of the molecular mechanism, how oral cancer develop metastasis is essential for the treatment in the future. And also the specific biomarkers and therapeutic targets to be validated in the future. So the situation in Thailand from in the past 2002 to 2004, there was a report of 1,186 cases reported worldwide that have a disease in Thailand, and with a 34.6% lesion in oral cavity. Data collection in 10 years from 2001 to 2010 in Thailand, in the northern Thailand to be specific, that the most patients diagnosed at clinical stage 4, with a 5-year survival rate was critically low of 27.4% for the patients older than 40 years old, with a major risk factor of tobacco smoking and alcohol consumption. So from the data collection that we can convince that oral cancer really remains major oral health problems in Thailand. From this diagram, at a histology point of view, you can see that from the left-hand side is a normal oral epithelium, healthy looking, but accumulation of the mutations and changes at cellular and molecular level over the period of times. The accumulation of this dysregulation of the genes has become, the cancer has become, the normal oral epithelium has become cancer at the end. And there are a lot of changes along the way. And what I'm interested in is that what really happened from primary tumor to become metastasis. So I believe that the biomarker discovery is essential for our understanding of what changes and what can predict and what we can predict before it becomes very aggressive. So the new knowledge is required for development of more specific drug target of oral cancer, and also for clinicians to have an effective treatment plan. So my role was to looking at the biology of cancer to be specific oral cancer, which is what I'm interested in. So we have a wide range of techniques and approach that we can take on to use to look at the cancer cells to be specific. On the right-hand side is 6 hallmarks of cancer that people talk a lot about. So what I'm interested in is one of these 6 hallmarks is the activation of invasion and metastasis, which is one of the crucial aspects of cancer biology. So today I'm going to highlight you with 4 basic research themes on oral cancer. First, starting with the genetic alteration of oral cancer cells. Number 2 is proteomic analysis of oral cancer cells, and also some functional studies to look at the specific genes. Number 3 is cisplatin resistance of oral cancer cells. We obviously have the knowledge that cisplatin is being used as the first line drug for that, but a lot of cases has evidence that the cisplatin resistance is a major problem for oral cancer therapy failure as well. And number 4, the last themes that I want to talk about is the biomarker discovery for the early detection, which I'm going to talk a little bit more about the microRNA, which I believe that our speaker, Dr. Mrna, also would focus on that as well at later on today. So this is our publication few years back, published on PLOS ONE. This is a genomic alteration found in head and neck cancer cell lines in the laboratory, and we did some karyotyping to look at the chromosomes and also some loss and gain of some genes that has been discovered from the approach of array comparative genomic hybridization, which I will show you later. This is a quite beautiful diagram of a multiplex fluorescent in situ hybridization karyotypes. So from these figures, you can see aberration of the chromosome numbers in the cancer cells, and also some translocation of the chromosome at different locations from the karyotype with a multiplex fish here. So from this figure, I can summarize you in this table with the different chromosome number and how many chromosomes are there in different cell line we have in the lab. So with this, we went on to look at specific location, at the specific location on the chromosome, whether which section of chromosome represents some genes that could be lost or gained. So we went on to use this approach of the array CGH here in collaboration with the Laboratory of Animal Cytogenetics at Thammasat University, and also Department of Pathology, Faculty of Medicine, Ramativati Hospital at Mahidol University as well. So we took our head and neck cancer cell lines and made a cell suspension, and then they extracted the genomic DNA in comparison with the normal cells as well. So all this genomic DNA was then labeled with a different photophore, which give different color. For example, here side 5 and side 3. And when co-hybridized on the slide with the target DNA, so then we then scan this to have this image, and then went to do the image analysis software. So we got the data at the end. So make the story short, we discover 4 important genes that has been linked to different locations across all the human chromosomes from the cell line in the lab. And there are 63, H19, WWOX, and PTPRD located at the arrow, as you can see from this diagram. And some of the genes has never been linked to cancer before. So we'd be quite interested to look at further and to do more experiment in terms of function. So also went on to check the expression experimentally of the 4 genes that we discovered as well. And some are upregulated, some are downregulated. So that would be the end of my first project that I would like to have an acknowledgement first here with Professor Sitichai Kuntongkao from Walailak University, International College of Dentistry, and also my co-researcher from Kasetsat University, the head of Kasetsat University Department is Associate Professor Khonsan Sikunat, and the team from Hidon University, Wellcome Trust Sankar Institute, and Biotech as well, and the funding from National Research Council of Thailand. So we went on to look at the protein. Sometimes when you want to study the proteins in the cells, we use the term of proteomic. It means that all the proteins in the cell that you're working on. So I extracted the protein from the cell. The same cell that I used earlier, the pair of the head and neck cancer cell lines. So we went to do 1D polyacrylamide gel electrophoresis, and then we did the mass spec analysis on this. Then we came up with the protein identification and research against the NCBI database using the mass cell search engine. And we found a list of proteins, which is quite interesting. On the right-hand side, this is the Venn diagram. So in the circle is actually a number of the proteins found expressed only in the metastatic cancer cell line, but not present in the primary cancer cell line. So one of the candidate protein I looked at and it came to my attention is a RISC protein. RISC is the ribosomal protein X6 kinase. It is a serine threonine protein kinase and constitutes a family of serine threonine protein kinase that are activated by the ERK-MAP kinase pathway. There are 4 members designated RISC1, 2, 3, and 4, and they share 73 to 80% sequence homology. So then I went on to look at the functional function of the RISC in the cell line. So the cell that I took and to be the model is a tongue squamous cell carcinoma cell line with lymph node metastasis, which we can inject it into the tongue of node mice and then determine this cell line as HSC3. Another cell line that I compared with is the HSC3M3. This is isolated from the lymph node of a node mice injected with the SSC3. So the HSC3M3 has a higher lymphatic metastatic potential with the same origin as the HSC3 cell line. So SSC3 and M3 has been used as the cell-based model to study the mechanism involved in lymph node metastasis of human oral SCC. And I used this pair of the cell line in the further experiments. So the objective of the project was to investigate the function of RISC1 in cell proliferation, cell migration, invasion in tongue squamous cell carcinoma cells and lymph node metastatic cancer cells. So what I tasted first was to look at how efficient of the siRNA transfection, which is the approach to reduce the expression of the genes in the cell. So I can reduce pretty much like about 70% comparing to the control cells. As you can see on the lower panel here is a Western blot showing the expression of RISC1 with the RSK1 siRNA transfected cells has a lot lower expression compared to the control. The first functional assay was to look at how cell can perforate in the cell that has reduced expression of RISC1. And we can see here what reduction of the RISC1 gene expression has a lot lower cell viability, and that was significant comparing to the control cells. So the RISC1 actually interferes with the cell viability of the cancer cells. Another things to look at whether how they can migrate and invade through the distant location, because when you look at the cancer cells and they are metastasized, they have the ability to invade through the basement membrane and travel through the bloodstream to distant location and form the metastatic cancer at very far away, distant from the primary location. So then I went on to look at the first assay. I look at how cell migrate using this approach. This is a live cell imaging. As you can see, I fast forward here. You can see the cell move to the gap, because I first scratch this to make a gap, and then I follow them over the 24 hours to see how well the cell migrate to close the gap. Okay, so basically, this is the experiment where I transfected the control siRNA with the RIS1 siRNA. 72 hours post-transfection, live cells were scratched and imaged for over a period of 24 hours. And then I measure the area of cell migration as a percentage of gap closure. So here's a representation of the image I took from both the control cells and the RIS1 siRNA transfected cells. And I got the result here of the live cell migration. Here I can see that they interfere with how cell migrate as well. The next experiment, I went how the invasion of the cells, whether infection or RIS1 can interfere with the invasiveness of the cancer cells. And the answer is yes, because when you look at the graph, the transfected cells compared to the control cell has a lot less cells, invasive cell, comparing to the control, and the effect was significant. So in summary, in this part of the project, we found that RIS1 inhibition affected both cancer proliferation of the tongue and also cell migration and invasion of a tongue HCC cell lines. However, how RIS1 is involved in the tongue squamous cell carcinoma metastasis will be investigated further. At this project, I would like to thank National Research Council of Thailand for funding and Professor Anand Arun, Faculty of Dentistry, Chiang Mai University, Thailand, for collaboration. Next, which is last part of this project, was to look at some specific molecule, which has been around some time and people really interested in, which is microRNA. MicroRNA have been explored in recent years as we have good understanding of their roles in cancer, also including oral cancer as well. I'm not going to talk in too much detail for that because another speaker will talk about that as well. So make the basically, they can control by binding to the mRNA target. Okay, sorry for the interruption. So I'll start this slide again. This is the mRNA processing. Basically, we have this in our cells. Everyone has this. So microRNA has the regulatory function. They can bind to the target mRNA. The result was either mRNA cleavage or translation being inhibited. So basically, in cancer cells, there's disruption of the expression of microRNA, either being highly expressed or downregulated. So the disruption of microRNA has been found to link with the diseases, including cancer as well. So what we have here, why are we interested in microRNA? MicroRNA has been found to be a potential biomarkers in oral cancer because there was a direct release of the microRNA from the oral cancer lesion in the oral cavity into the saliva, and also into the bloodstream as well, in the form of cell-free microRNA or exosomal microRNA as well. And also a release of the form of exosomal microRNA into the bloodstream and to the salivary gland, and then excreted into the oral cavity in your saliva. So we can detect the microRNA expression in the saliva and detect a specific microRNA genes of interest. Another story that people looked at is the drug resistant. In terms of oral cancer, we're really interested in the cisplatin resistant because, as I mentioned earlier, cisplatin has been found to is used as a first-line drug. So if you want to study this as a cell-based system, we can establish a drug-resistant cell line. For example, cisplatin-resistant cell line. So when we have cell line here, we can incubate the cell line with a drug, which can induce the resistance over a period of time. Repeated this incubation for a longer period of time will change the behavior of the cells, and some of the cells that's been selected can become drug-resistant cell line. And we can use this drug-resistant cell line to test a resistant mechanism in many aspects of the biological assays that you wish to test. And for example, you can use this cell line to screen for the drugs that's been claimed to test and fight against the drug-resistant cells, for example. So I took this figure from the publication of, I think it's quite good reports here about microRNA profiling in cisplatin-resistant oral cancer cell line. But they revealed distinct microRNA genes associated with cisplatin resistance. So this is quite a complex figure to look at, but there are a lot of microRNA genes here that they found linked to cisplatin resistant. And also they, on the left-hand side, they established the cisplatin resistant cell line to test of the further experiments as well. So I would like to bring you to this story because there was some data of my students that I have their name on the lower panel here. We found and we established the cisplatin resistant cell line as well. Like I mentioned, the SSC3 cell line. And we term the cisplatin resistant cell line as SSC3R. And we found that the IC50 are 10 times higher compared to the parental cell line as well. And we actually characterize this in terms of the expression of the biomarker that has been associated with cisplatin resistant, for example, vimentin and microRNA 2143P as well. Then we went on to test whether this resistant cell line has something to do with the RSK1 inhibition. And when I looked at a used inhibitor against this RSK1 inhibitor, we found that it affects the proliferation of the cell, this is a resistant cell line as well. So in summary, the cell viability of resistant cell line was shown to be affected by inhibiting risk activity. So this is a story that I and my students has been doing for the last couple of years. So recently, there are a lot of literature looking at the microRNA in oral cancer. So here I summarize the dysregulation of the microRNA genes at different anatomical locations, in basically focusing on head and neck region. And they found that there are different microRNA genes that has been differently expressed in different locations as well. So the genes that are underlined here is shown to be overexpressed. There are a lot of this, and they are different in different lesions in head and neck region as well. So this is quite inspirational. I would like to use these pictures to end my presentation. On the person on the right-hand side is Professor Aaron Tisanova. He is a Nobel laureate in medicine and physiology, working on the protein degradation, and he was an honorary lecturer in ISOVM 2015 in Zakopane, Poland. As I and my Professor Anap, which is the person in the middle of the picture, went together. And Professor Aaron said during the lecture that there will be no translational research without good basic science, then there will be nothing to apply, which is quite inspirational to me. So I think clinicians and scientists, if we work together, we can achieve a better project, and we can achieve something that I think is better not to work alone. And nowadays, I think I believe that people know that. So I would like to end my presentation here. So I think if I have some time left, I welcome some questions from the audience as well. Thank you.
Thank you, Dr. Ekarat, for a very observing and informative presentation. The platform is open for question and answer, so I request anyone who has any queries to please ask our speaker.
May I ask a question?
Yes, please.
Oh, thank you very much, Dr. Ekarat. That was very informative, very advanced, and emphasizing on this networking also, I think, is the most important take-home message for us, because I'm an endodontist. Obviously, I'm not in this network, but I used to work about some of the receptors ages ago about for example, non-malignant disease such as giant cell granuloma, central giant cell granuloma, or amyloblastoma. In the jaws, we have some disease that their cell lines is not malignant, but they are very invasive. And we need actually sometimes to work on them very radical. Do you have any idea, can we use this type of technology to evaluate these cell lines and compare those cell lines with the malignant cell lines and find it out what makes them very invasive? Because sometimes the type of the treatment that we need to do for them is very similar to the way that we deal with malignancy. Do you have any idea about working on the cell line of invasive but non-malignant disease?
From my experience, I'm only working on the quiet malignancies and invasive cell lines. So what I would like, in my experience, I would want to look at the mechanism, how they, because there are a lot of pathways involved that can drive cell line to be invasive or not. So there would be a lot of explanation on that, why they are the case. So, but I don't have a specific answer for that.
Yeah, so that can be a suggestion for the future work and collaboration between us to think about the way of, to find it out if a disease is very invasive, can we connect that, and can we use this technology to look at the proteins, to look at different types of the receptors and biomarkers and find it out, can we give an idea to the future for diagnosis of that? And also I have another question about the way that we treat when we decide actually, and we find it out, a cell line is very aggressive. Can it have some effect on our treatment strategy or not?
Do you mean the treatment, the drug, for example?
Yes, yes. I mean, for example, let's say about chemotherapy. Can we decide to do chemotherapy or not to do chemotherapy based on the result of your studies or similar studies at the clinical level?
At the clinical level, I'm not sure to give you the answer for that. But for at the laboratory level, we treat the cell. For example, I work with the cisplatin resistant cell line, and I used to treat the cell with cisplatin. So they are very sensitive to cisplatin on my hands. So it is the very invasive cell line, and I believe that is working fine on my hand. But in other clinical specimen, we need to basically maybe we need to compare the clinical specimen with the cell line in the lab as well, just to see the effect. Yeah.
Okay, thank you very much indeed, and I really enjoyed. Thank you.
Thank you very much.
Thank you, Dr. Nefokar, for your questions. We still have some time left, so would anyone like to ask any question to our speaker? Yes,
I
please.
think Professor Vipavi raised the hand.
Yes, please go ahead, Professor Vipavi.
Dr. Ikarat, if you unshare your slides, then we can
All
see
right.
all of the
Yes, sure.
participants.
Okay.
Professor Vipavi,
Yes, sure.
please go
Yes,
ahead.
yeah.
With
While we are waiting for Professor Vipavi to ask his question, to be honest with you, I want to ask another question as well, because I'm excited about the way and the technology that you use. My question
Yes.
about, can we use some sort of technique like that to look at the prognosis of a disease and also to predict such a things, to susceptibility as well? So for example, in people that are healthy, if we can do some tests and predict what is going on for them in 10 years or 20 years, because I remember that it was the connection between major histocompatibility complex, HLA, and susceptibility to disease. Is it similar to that? Can we use this new technology and advanced technology for such a things? Also, please tell us about prognosis as well.
Yes, I think I believe that microRNA genes is actually from literature. People looked at the microRNA genes. It's not only in cancer, but pre-malignant as well. So for example, like leukoplakia and things like that, they are associated with expression of the specific microRNA genes. And people looked at to basically the purpose of doing that is for early detection before it's become cancerous. So if we can do that, we can predict that whether these people will have likely chance and have a high chance to progress into cancer at later stage, like in the near future, things like that. I believe that is quite possible. Yeah.
Thank you. I think Professor Vipavi now is
Hello.
connected.
Yes, hello everyone. Thank you very much for the excellent lecture. My question is, as an oral medicine, I usually emphasize our students to early detect oral cancer. So my question is, just wondering if there is any biomarker that we can use for early detection of oral cancer. Is there any?
Yes, I think there's quite a lot of research on this such topic, but I would like to raise one of the hot topics on this is a microRNA that I just mentioned. That people looked at microRNA at different lesions and also in pre-malignant lesion as well. So I believe that is quite possible to screen for the microRNA genes at pre-malignant comparing to the cancer. So I think that there's a lot of research on that at the moment. Yeah.
Thank you.
Possible, yes.
Dr. Barak, may I have a question?
Yes, please.
Thank you. Thank you for your great presentation. I have a short technical question. Have you had any experiences to extract microRNA from formalin fixed paraffin embedded blocks? And would you share if you have any experiences to extract such microRNAs?
I have an experience of extracted microRNA from the cell, but not the embedded tissue yet. But actually, one of the project I'm planning to do that. So I don't have the answer for you right now, but I'm trying to figure it out soon. But I think it's possible to do that. That is quite a lot of technical protocol for that.
Okay, thank you.
Yes.
Does anyone have any other queries or questions before we move to the next presenter? No? Okay. Dr. Mohammed Kami, I request you to present the next speaker, please.
Okay, thank you. So I would like also thank Dr. Ikarat for his impressive lecture and speech. So, and now it's time to go to the next speaker, Dr. Katayun Sargaran, who is my dear colleague in the Department of Community Oral Health in School of Dentistry, Tehran University of Medical Sciences. Dr. Sargaran got her DDS degree in Azad University in Tehran and got her PhD degree in University of Helsinki in Finland. And her field of research and interest is oral cancer prevention at community level, the area that she wants to share some of her knowledge and experiences with us now. Dr. Sargaran, the floor is yours.
Okay, good morning, ladies and gentlemen. I'm very happy to be here with my colleagues from TUMS and Thammasat University to present aspects on community and individual strategies in oral cancer prevention. As a short introduction, the global cancer surveillance in 2020 showed that 19.3 million new cases of cancer and almost 10 million cancer deaths is recorded and estimated, which is 1.5 times from 2008. As you can see in this world map, Iran is one of the prevalent cancer cases countries, but we are going to go through oral cancer, which is not too much prevalent, but we know the consequences, of course. Oral cancer ranks 18th among all cancers combined and comprises 2% of all new cancer cases and 1.8, around 2 of all new cancer-related deaths. As you see, Melanesia and Indian subcontinent are still the most prevalent areas of lip and oral cavity cancers. And the mortality rate is also very high in these countries. To point to Iran, to come to our country, it is around 2 new cases, age standardized incidence rate, and almost 1.3. Which you can see in the graph in Western Asian countries. The major risk factors are, as you know, alcohol drinking and smoking, tobacco smoking, chewing, or using in any type, and the estimated attributable risk of smoking and alcohol consumption is around, for the population, of course, is around 80% for smoking in men and 60% in women, come to almost 74 when we look at both genders together. And about the increasing in the ward figures, ward prevalence, incidence rate of oral cancer, it can partly be due to aging of the population, but also the high or increasing levels of risk factors, which are smoking and drinking, as I told before. But in some countries, any types of using of tobacco in different shapes. And also it can be, as you know, due to the higher usage of smoking in the women and in youth. Sorry for the interruption. Community preventive strategies can be easily adopted for the prevention of oral cancer, because we have several initiatives, global initiatives, working on these preventive strategies, recommendations we have. And also oral cancer is still a silent epidemic, it is called actually. And this is because there is not enough attention of policymakers and administratives in this regard. But it is potentially one of the cancers which is potentially preventable. So it should be it should mention in the level of prevention. Okay, as you see in this picture, in this photo, that regarding the level of prevention in different phases of onset of disease, we can have the different preventive strategies. For example, in the preliminary or primordial phase, which we don't have any disease, the prevention strategy should focus on the development and to ban or combat the development of risk factors. For example, it can be through setting different regulations and imposing or raising taxes on the different types of alcohol or smoking products. As you see, this is a high-level upstream strategy which should be prioritized in the policymakers and in the health systems. Another level which still we don't have any disease, any onset of disease, is the primary prevention level. In this phase, the target is to manage the risk factors, and for example, by public education, and it can be also, the public education can be used in the primordial prevention level, but for example, for the younger adults and for the adolescents. But in the prevention, primary prevention level, as I told, the management of the risk factors is the main goal. So it should be, for example, to adopt policies in quitting the smoking or cessation, and also for alcohol drinking, of course. The secondary level, in this level, level of prevention, the disease actually is now present, but it may be asymptomatic. So the main goal will be to detect the lesions, pre-malignant lesions or malignant tumors in the first stages, in the early stages. And so the strategies mainly focus on the empowerment of the professionals for making the early detection and diagnosis. So it's also related to the prognosis of the disease. of course. Another level of prevention is the tertiary level, in which we have the clinical course, and the clinical diagnosis is now, but we have to focus on the reducing of clinical complications and consequences of the disease. So it is related to the treatment and rehabilitation, of course. So you see that in any of these levels, 4 levels, we can intervene and have the strategic goals. About the regulations and legislative measures, we can say that the policies and regulations strengthen the governments and non-governmental organizations such as NGOs and other administrative authorities to act for reducing the incidence and consequences of oral cancer. Some of these regulations and legislative measures I have listed here for you, such as banning the tobacco and alcohol use to ensure adequate legislations or bylaws for the lower level administrative authorities, and to ensure warnings on the product sold, as of course in many countries, almost all around the world, we have these warnings on the cigarette packages or on the alcoholic beverages bottles, but we have to see what are the consequences of these, of course, and also to increase the cost of these products and to reduce the cost of other healthy substitutes, and also to avoid the glorification of the products or the use of the products through advertisement or media, mass media, series, cinema products, films, something like that, which is unfortunately very prevalent these days in the most popular series, which are mainly seen by our adolescents or by the young adults, all the population. So these were the regulations and strategies from higher level. Another strategy will be the screening. The major question in this regard always have been that it's recommended as a national whole population strategy or not. First, I have to say that screening can be like whole population screening or opportunistic screening of high-risk groups. Both of these strategies have, of course, limitations. For example, for the whole population screening, there is still no sufficient evidence. I mean, in the countries that oral cancer is not so high, please be aware that I'm not talking about the Indian subcontinent or the other areas of high prevalence of oral cancer. In those areas, whole population screening is highly recommended. But for other countries, there is no sufficient evidence on the costs, on the benefits, cost-benefit, cost-effectiveness, and also the feasibility and appropriateness of this screening strategy. Another one, the opportunistic screening of high-risk groups. The main limitation for this, that when we see those who are at the highest rate of risk, which are older men who are heavy smoker and drinker, they are not attending as a regular basis to dental clinics. So this cannot be applicable. So these are things that should be thought about when we are speaking about the screening. Another strategy, which is mostly acceptable, of course, are the community-based health or oral health promotion programs. These can be very successful, but the understanding of underlying social, economic, and political determinants, and also the barriers, is very important when we are designing these types of programs for the population. The most important thing is that these programs should target both high-risk groups and those who suffer health inequalities. And any of these two as a sole strategy cannot be actually acceptable and cannot come through success in the community programs, because the most people who need these types of awareness or these types of intervention are those who are both in the high-risk groups and also those who have the health inequalities. As we know, oral cancer is 4 times more prevalent in underdeveloped socioeconomic levels. So this can be a goal target in these programs. And also another very important option that we have to bear in mind is the common risk factor approach, which these programs should be integrated with other health promotion programs targeting cancers or other non-communicable diseases which have or which share the same risk factor. Coming through these community programs, we have 2 main keywords. One is the common risk factor approach, and another is the integration. So to come to a conclusion in the health promotion activities, it should be noticed that utilizing a range of health promotion activities, we should act beyond the sole reliance on the health education and improving the public awareness also, although it is important actually, but the strengthening of the community, developing personal skills, and also enabling advocacies, creating supportive environments, and reorienting the health system in this action is very important. So as I mentioned before, the advocacy and the networking of all the health professionals is important in this regard. Global initiatives are also very important for all the countries around the world to strengthen these the policymakers or global activists, government, or other health system professionals, and they are especially those in prevention of oral cancer and tobacco control. I named some of these important ones, which are the Threats Declaration in Oral Cancer Prevention, which was launched in 2005, WHO Framework Convention on Tobacco Control, which is famous as FCTC, from 2003, and also the Bloomberg Initiative to Reduce Tobacco Use since 2007. It is an ongoing project. And also the World Assembly report on oral health, including cancer prevention, which is from 2007, and it is ongoing every 2 years, if I'm right. Okay, about the CRET declaration, the main points of this document are the active involvement of oral health professionals in collaboration and in cooperation with other health professionals in the health system, as I mentioned before, for the prevention programs. And the other topic is the access of the public to the facilities for the early diagnosis and detection of different lesions. So it needs the networking, team working, and the areas which are needed the most strengthening, according to my opinion, in our own country, of course. In the WHO Framework Convention on Tobacco Control, it asserts the importance of demand reduction strategies as well as supply issues. And it recommends some price measures and non-price measures. Price measures, I mean the strategies regarding the tasks and costs, but non-price ones I will name in the same in the next slide. For example, regulations for packaging and labeling, education, communication, training of the public, and demand reduction measures concerning the tobacco cessations for the empowerment of the dental professionals and other health professionals in this regard. In the Bloomberg Initiative, the philanthropists have committed nearly 1 billion since 2007 to combat tobacco use worldwide through different methods that combine policy change with public awareness. And it is very important to see these two together. As I mentioned before, policymakers should be involved in all of these strategies, not just giving the public awareness solely. It also uses the Empower named framework to fight tobacco use across the globe. It is recommended, and the Empower stands for monitor tobacco use and prevention policies, protect people from tobacco smoke, offer help to quit tobacco use, warn about the dangers of tobacco, enforce bans on tobacco advertising, promotion, and sponsorship, and raise taxes on tobacco. Many countries use this framework in their strategies, and many success rates come through. Also, we have it in our country. I will go through it in the next slide. Okay, in Iran, the comprehensive law on national tobacco control launched from 2006, and we joined FCTC, WHO FCTC. We were one of the countries that signed that report, and according to that, countries should have regular reports on their actions. And Iran has used the guidelines for strengthening and supporting the governmental and non-governmental actions to tobacco control policies. And as I told before, a bylaw which targeted the banning of tobacco product consumptions in all public places and workplaces, public transport, and outdoor public spaces was implemented after 1 year, and the comprehensive measures have been taken to adequately enforce the smoke-free legislations. And also, these were resulted in additional educational advocacy activities that have reinforced tobacco control efforts in Iran. But actually, we have a long time through the ideal success in all of these areas, because as we see in any other country in the world, the trade marks and the market is very important and have the most strong strategies for their own, of course, to combat these health strategic plans. Okay, to come to individual level preventive strategies, these strategies also can be used in the clinical setting all over the countries. Dentists are the main professionals in this regard. Also, patients can be involved actively in these prevention strategies. To come to patient-centered strategies, the main goals are to improve the demand of the patients for preventive services. Actually, you know that most of the dental patients are those who coming to our clinic for treatments and not just for preventive services. So this is the responsibility of the health system professionals, dentists, to make this, to improve the demand for the preventive services. And this is not possible just with doing the treatments, but we have to give them the opportunity to be aware of the positive effects of preventive services. Another is the periodic regular visits of the patients. If they do this, one of the most important barriers of the oral cancer will be, of course, omitted. And the avoidance of known risk factors, as I told before, can be also focused in the patient-centered strategies. Self-examination and referral is something which is now very in trend in the different health professionals' activities, and it should come into important different health promotion programs also, not just in clinical centered ones. And the dental service utilization early and on demand should be focused. So as I mentioned before, to improve patient awareness of these are very important. And also we have to take into account that people usually in many countries, according to their cultures, for example, in Iran, we have this problem. They have a psychological problem that is fear of cancer, of carcinophobia. In this regard, patients can be in two different types, actually. Some are very some want to use the dental visits several times, and it is somehow over regular visits we see in these types of patients, and the others are delaying the periodic regular visits. So dental professionals should be aware of this and to know how to manage these two types of patients actually. Another individual level strategy is the professional based, as I told. Patient education, patient counseling about the risk factors and risk factor assessments in every visit, every checkup of the patients is very important in this regard. Comprehensive oral and extraoral examinations should be done. I think in 1999, WHO have an initiative in this regard and says that oral cancer prevention. By 5 minutes examination, you can save several lives. So we have to educate this in our undergraduate dental education and to focus on this more, I think, and to attempt in early diagnosis and the referral. So in this regard also we have several problems, because the most oral cancer cases in Iran diagnosed in late stages, more than 30% of these cases diagnosed in stages 3 or 4. And it is important to have prompt referrals and to referral to a specialist who knows what to do in this regard. And also proper biopsy, cytologic examinations, and excisions also should be noted in this aspect. And proper, to come to a conclusion, proper professional teamwork and networking in undergraduate and postgraduate specialist networks, and also different specialist networking should be performed in different countries. Yesterday, we discussed these points in the previous webinar with our colleagues, and they will agree in this regard that our undergraduate students shouldn't lose the connection with the university after they graduate. So to promote early detection and to reduce the fatalities from the later stage diagnosis, and also to improve the quality of life of patients and also the survival rate, both patient and professionals are responsible, not just the patient and not just the professional. So in the delay, we have patient and professional delays. Patients come late, professionals diagnose and refer late for the treatment. So in diagnosis and in the referral and treatment, both, we have to reduce the delays in oral cancer to improve the survival rates of our patients. In conclusion, I wanted to conclude the community-based strategies targeting the most important ones, which are building healthy public policies, tightening restrictions on tobacco and alcohol sale, advertisement to subsidize the costs on healthy choices like fresh fruits, vegetables, alcohol-free drinks, and to improve the labeling, awareness labeling, warning labeling on the related products. And also to create a supportive environment, not just for the population, but also for the professionals, in order to have smoke-free spaces, cinemas, public transports, etc. And availability of healthy products should be in all schools, in chain markets and small shops all through the country. And also to strengthen community actions, for example, using the health volunteers capacity in this regard to promote establishment of local community-based smoking cessation support groups, to establish helplines, telephone lines, for example, or internet sites that appeal to population groups with high rates of smoking or drinking, and support the establishment of local food cooperatives selling cheap, high-quality fresh fruit and vegetables. And also to develop public personal skills in personal care, of course, to expand personal and social education in different places such as schools, and to empower people in life skills, their own life skills in the psychological properties such as refusal and negotiation skills, and to incorporate tobacco and alcohol control within health promotion, promoting school initiatives. And to, at last, but not the least, the reorientation of health services to expand health professionals' education and training in smoking cessation and alcohol control, in early diagnosis and prompt referral, and to increase number and range of health promotion professionals and experts to establish evidence-based smoking and alcohol preventive services within primary care setting. Thank you for your attention.
Thank you. Thank you very much, Dr. Sargon, for your informative lecture regarding various initiatives, interventions at community level to prevent oral cancers. So I think we have, yes, we have 5 minutes for question and answer. So if there's any question from the audience, please just raise your hand or open your microphone and ask your question. Okay, Professor Vipavi, sorry.
Please unshare the slides.
Okay.
Hello.
Hello.
Hello. Thank you very much for your informative presentation. I just wondering if beta nut chewing is also a problem in Iran, because in Thailand, besides smoking and alcohol drinking, we also have problems with beta nut chewing, especially in northeastern region of Thailand. Many people who suffer from oral cancer, they are beta nut chewing people. So how about in Iran? Do you have problem with beta nut chewing as well?
Okay, beta nut chewing is not prevalent in all the parts of the countries, but in 2 states in the east part of Iran, we have some kinds, and also we have the problem of using similar somehow products like beta kit.
What is that?
But it is not all over the countries. It is called some NOS in our language, and
oh,
it
yes,
is not
right,
exactly
right. I heard that.
Yeah. It's not exactly like the betel, but it's a mixture of tobacco with other things, and they use it regularly, of course, in those parts actually, but not in other parts of our Iran.
And is that also associated with cancer, oral cancer?
Yes, oral cancer are more prevalent in those parts of Iran, in Sistan and Balochistan, and north of southeast of Khorasan state.
Okay. Okay, yeah, thank you.
Thank you.
Okay, thank you very much. In total, just less than 5% of tobacco usage in Iran is related to chewing tobacco. So it's not high, as Dr. Sargon explained, it's about in some parts of Iran. And then we have another question by Dr. Siriwan.
May
yes,
I? Yeah.
Siriwan. Yes.
Thank you, Dr.
Please.
Saran, for a very comprehensive model for prevention of the oral cancer. I just would like to know the role of the village. I mean, in Thailand we have village health volunteers, you know, to help on the preventing the oral cancer or other oral problems. For example, in Thailand, we have the program. I think they are the group from inter-country for oral health in Chiang Mai province in the north of Thailand. They have the model that they try to train, the village health volunteer to diagnose. Just to check the early detection of the oral cancer. They develop the simulation, the very simple simulation, so that they can train the village health volunteers to be able to detect the oral cancer lesions and then go to the referral system. Do you have this kind of idea in your country?
Actually, we don't have the program as you have because we don't have this prevalence of oral cancer in every remote rural areas. But in some parts of Iran, I know that we use the help of health volunteers and health workers in PHC service system, which is covered all over the country, to improve patient awareness and public awareness in this regard, especially in smoking programs, targeting not just only the oral cancer, and it is not included the examination, of course, but just the working on the knowledge and regular dental visits of people in this regard.
Thank you.
Thank you. Thank you very much. So, okay, so I think, thank you very much, Dr. Sagran, again for your nice lecture.
Thank you.
And now I think it's time to go to the next presenter that Dr. Baharat will introduce.
Thank you, Dr. Kami. So it's my pleasure to introduce our next speaker. He's young and energetic. He finished his residency training program in oral and maxillofacial surgery at University of Khon Kaen. Currently, he's a lecturer in Department of Oral and Maxillofacial Surgery and also a diplomat in Thai Board of Oral and Maxillofacial Surgery. His field of research interests include artificial intelligence and deep learning in oral and maxillofacial surgery. So please welcome Dr. Kritsasit Warid.
Good afternoon, everyone. Good afternoon from Thailand. Can you see my slide?
Yes, doctor, we can see your slide.
Okay, let me introduce myself. My name is Kritsasit Warid. I am the oral and maxillofacial surgeon in Faculty of Dentistry, Thammasat University. Okay, I would like to thank for inviting me to this interesting event about oral cancers. Okay, today my presentations today I have two presentations. For the first one is about the assessment of the oral cancer in the term of the clinics. And I would like to share my specialized to the audience about the oral cancers. Okay, for the first one, I would like to for the survey worldwide, the oral cancers is the 18th most common cancer as we know, but in my region, it's the fourth most common cancers in Southeast Asia. In my country, Thailand is the 10th most common cancer, and it's found mostly in the northeastern part of Thailand. I would like to share everyone that maybe everyone know oral cancer only for the oral squamous cell carcinoma, which is most common of the oral cancers, but it's only the epithelial tumor, epithelial origin tumors of the oral cancer is just one, but the other 10% is including malignance of the salivary gland tumors or the pigment oral lesions, which is the oral melanomas, or the hematologic malignant tumors that it can show in as the oral type, one type of oral cancers. Not only that, one of the most aggressive one of the oral cancer, which is malignant from the bone tumor, is this osteosarcoma, this picture is my case of the osteosarcoma at the maxilla. The other is the substitute sarcoma, for example, fibrosarcoma, liposarcoma, which is originate from the fibroblast or the lipid tissue, something like that. Or we need to recognize and realize that not only the cancer in the oral cavity is come from the oral cavity, but it can metastasis from the other regions, such as the thyroid carcinoma, which is can metastasize to the in our oral cavities. For the risk factor, okay, for the risk factor of the oral cancer is normally, as we know, that is tobacco use, between chewing, alcohol drinking, or the high fat or sugar intake, or the infection of the human papilloma virus or HPV virus. But in Thailand, there is a research and survey in the oral cancer screening program from the 4 provinces in the northeastern regions of Thailand. The research of the Professor Dr. Bawon Khongnoi, Mahidol University. For the risk factor in Thailand, in the highest prevalence of oral cancer, which is the northeastern Thailand, is the first one is alcohol consumption, the next one is smoking, and this one is the one of the most important risk factor in our region is the between consumption or between chewing. As you seen in my case of the oral cancer, the strain, the black strain in the on the teeth is all this is with straining, which is show that the patients regularly has the with chewing. Oral cancer, as I already mentioned, is the most common oral cancer. 90% of all oral malignancy is the OSCCs. Mostly there's this type of cancer as shown in the clinics in the three features. The first one is the ulcerative lesions. The second one, exophytic, and the last one is endophytic lesion, which is maybe difficult to recognize and difficult to examine if the clinician has no or less experience in the oral cancers. Before I speak about the clinical examination of oral cancer, I would like to share about the pre-malignant lesions, which is currently called oral potentially malignant disorders or OPMDs, which is the mostly of OSCC is transformed for these OPMDs. So the early detection of the malignancy lesions, the best is we can recognize it before it's turned to malignancy lesion, which is the pre-malignant lesion. But as we see in the picture, maybe it's something like very difficult to recognize and regularly misdiagnosis by the clinicians and lead to the patients to the late diagnosis of the oral cancers. As we know, okay, this is a sample of my cases. The first one is the retromolar dicone. The right side is the OPMD lesion, and the left side is the oral cancers or OSCC lesions of my cases. As you see, maybe in the retinal tongue lesions, you see in the erythematous lesion or the red lesion, but normally this lesion as patients have no any symptoms, and maybe if they come to the dental clinics and the dentist did not see it and miss for the diagnosis, maybe it can lead to the left side of the picture that's developed into the oral cancers. Okay, let's see. It's about the assessment of the oral cancer. In terms of the treatment of the clinical evaluation, the first one, we need to have completely the history takings of the patients, including analysis of their risk factors. The second one is physical examination, general medical examination of the patients. And next, if we examine the patients truly, then we send for the imaging and including with the biopsy examination for the pathological result. For the physical examination of the oral cancer, okay, we are the dentist or the oral cavity specialized, but we need to see the patients truly. The physical examination not only include the oral examination. We need to examine all of the general health of the patients, especially for the cancers. The cancers may be not only manifest in their oral cavity, but maybe have the metastasize into the other organ, or for the head and neck region, we need to not only examine for the oral cavity, but we need to examine the head and neck region truly. For example, one of the most important part is the cervical lymph node examination, that we can examine for the clinical, that as if any metastasize of the cancer to the lymph node in the next regions. And then we see the oral cavity, the lesions inside the mouth. Because we are dentists, it's normally we can we have the specialized in the oral cavity. We know of the oral cavity, but mostly from my experience, as I see my juniors, most of the general dentists miss in the examination, in the extra oral examination, especially for the head and neck or the head and neck region apart from the oral cavities. That may be that may lead to the missed examination for the patients. Okay, the next one is the imaging. After we exam for the physical examination truly, so the next one is we select for the further investigation. For the first one is the easy one is the imaging from the plain film, such as the polypical film, occlusal view, or the panoramic view. That we can access. No, okay, as we know, the plain film maybe can't access for the substitute lesions, but it can access for the bony invasions. And the next one is the CT scan or MRI, that we can evaluate for the tumors, the truly or from the all pains, both of the substitute and the bony invasions. And then if we suspect for the lesions, that the pathologic lesions, the pre-malignant or not, or the cancers or not, we need to confirm with the pathological examination by incisional biopsy. I suggest that if we suspected that it may be the cancers, we should not perform the excisional biopsy. We should just the incisional biopsy to confirm for the lesions, because the treatment of the pre-malignant and the malignancy lesion is really different, and the malignancy lesion is very more invasive treatment. And I will have my cases for example for the next part. And then after that, if we have the pathological examination, pathological results that the patient has the cancers, and the next one we need to treat the patient. For the first part is the treatment planning. For the treatment planning, we need to evaluate the patient truly. For the first one is the pre-operative assessment. We should not miss the general health issue, because we are the dentists. Maybe we know mostly in the mouth, but in the general health, this part is one of ours, not the specialized, and we are easily to miss the information of the patient general health. The first one is history taking. We need to see that in general before the other part of examination, the physical fitness. We can evaluate the patient. If patients can walk, if patient can do any daily activity with the normal or not, or any illness or any origin of anything we need to evaluate. For the physical examination, as I mentioned, not only for the head and neck region, we need to evaluate for the other regions such as the heart. We need to listen for the heart sound. If the patients have any murmurs or any heart disease, or for the lung sound, if the patient has screening for as if the patient has any abnormal in lungs in the physical examination, and then we send for the further investigation, such as the laboratory investigation in the blood, laboratory investigation such as the CBC, complete blood count during analysis, BUN, creatinine electrolytes, liver function test, and anti-HIV, or the examination for the chest X-ray or the heart examination is from the electrocardiogram or ECGs. Okay, as I mentioned for the preoperative, okay, after we had the imaging such as the CT scan, I suggest for the malignancy lesion in the oral cancer, I suggest the CT with contrast, because with the contrast, we can evaluate for the lymph node metastasis from the next regions. And for the CT, as we know, the CT has the two windows, normally two windows, the soft tissue and bone. We need to evaluate both of the soft tissue and the bones of the patients, as it weighs the how large of the soft tissue lesions and is there any the bony destruction from the CT that we can evaluate the how aggressive of the lesion of the to the patients. Next one, if MRI is, as we know, that is the best one for the examination for the soft tissue lesions. The next one for the chest X-ray or the chest CT for the some patients that we can exclude for the lung metastasis, because as we know, one of the most common site of metastasis from our oral cancer is lung. The next one, the ultrasonography. Maybe for the first one, for the ultrasound sonography, we can evaluate from the next region, we can evaluate for the cervical lymph node metastasis, or we can see the liver, is there any liver mass? If there is any liver mass with the oral cancer, maybe we choose further investigation is under liver metastasis or not. The next one, if some center can, I think this one is one of the best evaluation for the tumor metastasis now with the PET-CT but for some center maybe this evaluation may be difficult to sense not by okay. And the next one, if you suspected there is any bony metastasis, you can sense for the bone scan or bone scintigraphy. The CT one, yes, there's this example of the CT of the cancer case. This one is the soft tissue windows that we can see the lesions, but okay, but the soft tissue, some of the soft tissue windows we cannot see the bone clearly, but we can see that this one is the has the sum of destruction of the alveolar bone in the mandible. Okay, the next one, after we have the sense, we have the result, we have the examinations. Okay, the next one is the surgical size. Okay, we need to evaluate is this cancer need the surgery or not. For the surgical side, we need to evaluate for the tumor. What's the tumor extension? Is it resectable or not? Resectable or not is the tumor invade to our vital structure, such as the blood vessel in the neck, for example, internal carotid artery, or the vesicle or not. Is that something we can't dissect it completely? This is something we call the unresectability of the tumors. The next one for the next, yes, we need to include all the clinical examination for the lymph node. Is there any lymph node metastasis combination with the imaging for the CT? Yes, is there or the ultrasound is if we have the information for it. Is the patient has the suspected for the cervical lymph node metastasis or not? This one is the very important information for the treatment planning of the oral cancers. And the next one is the reconstruction of after we have the surgery, after we remove the tumor. After we remove the tumor, as we know, the tissue, the bony tissue, the soft tissue is lost along with the tumor. And after we remove it, we need to close it, and we need to evaluate. It's something like the donor size for the flap or grafting to close the defect of after we have the surgeries. Okay, next one, we evaluate the staging of the tumor. Currently, especially in my country, we evaluate the oral cancer staging with the American Joint Committee on Oral Cancers, or OJCC. Currently, the TNS staging use the 8 editions. The TNS staging of the oral cancer, we need to evaluate the three part, and summarize it as the stage 1 or 2 or 3 or 4. 4 is divided into A or B or C. The first one, we need to evaluate the size or characterize of the primary tumors. The next one is the regional lymph node, and the last one is the M or distant metastasis. Is there any one? And we conclude summarize for the staging of the patient before we start the treatment. Okay, the next one is the select for the treatment of the oral cancer. The first one depends on the size. As we know, in the oral cavity has divided into 7 or 8 regions: upper gum, lower gum, for example, upper gum, lower gum, or the tongue. The different size has different treatment planning, because after we cut it, we remove it. How we close it? Yes, this is the one of the challenge, the size. Or the size is very important. Another one is different size in the oral cavity have the different prevalence of the lymph node metastasis. The next one is the staging. The next one is the histological part. This is the oral cancer or OSCC. What this is differentiate or what this is characterized: the poor, moderate, or well. Next one is the patient compliance and general health. From my experience, we need to check for the patient's compliance, because we need to tell the patients what is patient had, what the cancer, and what is the treatment after this. If the patient chose for the surgery, we need to tell for the complications or the side effect of the treatment, and the compliance of the patient for the post-operative care. If the patient accepts for the risk and or anything, we can go on for the surgical treatment. If not, I suggest that we can propose for the next treatment option for the patients. And next one is the surgeon's ability and team and faculty. This one is very important, because the surgeon has the different specialized and different ability for the treatment of the oral cancer. For example, some surgeons cannot perform the neck dissection or remove the lymph node at the neck of the patient. The next one is as we know, the oral cancer is the multidisciplinary issue. We need to have the tumor conference between the surgeons, radiation oncologist, or medical oncologist for the treatment planning of the patients. Okay, the treatment of the oral cancers, maybe I can conclude. Normally, it has, I think, it has 2 options. The last option, the major option, the first one is the surgery with the combination of the radiotherapy or chemotherapy. The next one is patient should not have any surgery. They want only the radiotherapy or chemotherapy. Something depend on the state. Some state or early stage, only the radiotherapy is okay. But for the late stage, maybe it's called something like the palliative treatments for the patients, or the additional therapy that we need to realize and aware for the. Then this one is very important for the general dentist, is the oral care before and after radiochemotherapy, because many of my cases come to my department, maybe refer from the hospital, the other department, for example, the plastic surgeon or the ENT surgeons. They have already performed for the oral cancer surgery, but did not prepare for the oral health. The patient come to me with the generalized chronic periodontitis, and we need to extractions many teeth of the patients, but the patients already had the radiation therapy. So it's a very difficult decision for perform any treatment, because if we had any surgical management in oral cavity, especially the tooth extraction after the patient had the radiotherapy, maybe we need to tell the patient for the other complication, which is the ORN, the osteoradionecrosis of the jaw, which is the one, I think, one of my most test complications of the oral cancers, because it need very, very long time for the manage this one of patients. And then we should not forget about the rehabilitation of the patients. After we cut their structure, we need to, after we replace, but as we know, our oral cavities has the functions, the chewing, the eating, the swallowing, and speaking. After we remove all the tumor, we need to realize about the rehabilitation to the patients, to have the patients have the best quality of life for them. For the prognosis of the oral cancer, I would like to share one of my research. It's published in Thai language in the Thai Journal of Oral and Maxillofacial Surgery. I analyzed for the 5-year survival rate of the patients in my training center in Khon Kaen University, which is in the northeastern part of Thailand. We found that most staging of the patients in our centers is as you see in the table, the stage 1, 2, or 3. The stage 1, then okay, maybe the result is something. Most of my patients is the stage 3 or 4, and very little number of the small number of the stage 1s, because in Thailand, most people, as I said, most people has low awareness of their oral lesion, so they come to see the doctor after they had the large lesion. So most of my patient is the stage 3 or stage 4. So the 5-year survival rate of my study maybe something like stage 3 is now affect the survival rate more than stage 1. Is from the number of the patients. But not only my studies. When I compare with the other study in the different parts or different countries, we found that the best prognosis of the patient is the early stage, and for the late stage, especially in the stage 4, as you see, it has very poor prognosis, and the 5-year survival rate is only 20%. And overall 5-year survival rate, I can conclude that maybe for the oral cancers, that only the 30 to 75% is not so high. So we need to realize how important of this disease, because if I compare the 5-year survival rate of the 30% is something like if the patient come to the patient with the oral cancers come to my department, the 10 patients, maybe after the 5 years, the patients will remain only the 3%. Okay, I would like to share my oral cancer case. For the first one is the 47-year-old Thai male that come to my department, had the lesions in the lower gum for 2 months and is rapidly increased in size. He's already come to, before see me in my department, he already go to the private clinic, and the dentist that he found, maybe general dentist, they diagnosed him as the chronic periodontitis or the swelling, the gum infection, something like that. So he had only the antibiotic with the amoxicillin and the anti-inflammation medicines, which is the ibuprofen. And after that, he told me that the lesion is not subside, but it's large and larger. And then he come to see me at my department. And after I see him, this is the picture of his lesions. in the lower gum and is already invade the his floor of mouth. The pathological result that after I had the incisional biopsy is the OSCC with the moderately differentiated. When I have the other examinations, the conclusion is the staging of the is the T4A, N1, and M0 or stage 4A. Yes, as you know, he come to see me as the stage 4 or the last stage of oral cancers. This is maybe it's something like daily life in Thailand that the patient has the very low awareness, or something like they have awareness what is there any abnormal in their mouth, but the general dentist has maybe have the low indicate about the oral cancer, so they miss the act of it. The treatment of these patients, the first one I perform for the Referral selective neck dissection. For the last one is I can palpate for the next node metastasis. So I perform neck dissection for the level 1 to 4, and the left one, because of the tumor is across the midline of the mandible, so I perform the left side with the level 1 to 3. After that, I perform the wide excision along with the segmental mandibulotomy. And this is normally for the OSCC, we need to cut with the surrounding margin around 1.5 to 2 centimeters. That's the call is something like we see it. Okay, we see it. It's the feature at this one, but for the surgery, we need to include all other surrounding, normal surrounding around 1.5 to 2 centimeters. That's in these patients, I cut almost from the angle of mandible to the angle of mandible of the patients, almost all of the mandible of the patients. And after that, I use, because of the different prognosis of the tumor of the stage 4A and the tumor characteristic is the moderately differentiated. I discuss for the patients for the treatment option for the region, for the local flap or the free flap, but the patients choose only the local flap. So in this one I use the reconstruction plate to replace the missing mandible and close the substitute defect with his remaining tongue is the tongue flap, as you see as this picture. This is for my first case that I want to show you that if a patient has the oral cancer, we need to realize after the treatment its impact for his or her quality of life for sure. This one has no tooth remain. So and he choose not have the free flap. So the prosthesis is maybe it's not the choice for the future. So after the surgery, maybe the remain of his life maybe he can eat only for the soft diet. He can't chew or anything. But if he choose for the free flap reconstruction, after this is the other issues. The other one I would like to share is the case is of my case is the oral melanoma. Come to the department is a 69 years old Thai female present with the black lesion in her mouth for 4 months and is gradual increase in size, and she comes to my department. This case is that I have the publications already have the publication. You can go to read for the full papers. I perform for these black lesions in incisional biopsy, and the pathologic result is the superficial invasion of oral melanoma. As for the dentist, maybe the black lesions that we familiar is something like amalgam tattoo. I don't know if that Iran currently had the amalgam filling or not, but in my country, some patient has already currently has the amalgam fillings in their mouth. So some of the dentists may be recognize black lesion in the mouth as the amalgam tattoo or any nevus or something like that. But they did not aware that the black lesion in the mouth is one of the most aggressive oral cancer types, is the melanoma. In this one, maybe I perform the biopsy, the pathological result is shown as the something like superficial invasion as you seen from the pathological image. But after I sent for her for the CT scan, this one is show that it's not superficial invasion as shown in the pathological result, but it has the bony destruction. It shows that melanoma is very aggressive tumor, and it has the poorer prognosis than the OCC. This one, as you see, for the treatment options, I propose for her that the first one is surgery along with chemo or radiotherapy. But the next one is has no surgery, only the palliative chemo radiotherapy. And she chose for the second one in this case. Because of as we know, the oral melanoma, maybe after we perform the surgery to reach, but the prognosis is not so good. So the early detection from the dentist or the physician is very important. This one is the one example of my case that I already published. Okay. For the conclusions, I want to tell that as we know, the oral cancers, the early stage has the best prognosis and the best and the highest of the 5-year survival rate, and is the lowest is in the late stage, especially in the stage 4, as we seen in this table. As we seen some of the studies, it showed that maybe the 5-year survival rate of the stage 4 maybe less than 20%. So it's very bad prognosis. So the most important part is the early detection. For now, maybe for all of the current treatment of the oral cancer, as we know, the 5-year survival rate is not improved for many decades. Maybe the best for patient is the early detection to manage in the early stage or just the pre-malignancy lesions. So this one is why I'm interested for using the computer vision technology or artificial intelligence for use for the health using for the oral cancer detections. Okay, this is of my first sessions of the oral cancer. For the next session about the artificial intelligence, I will tell you for the next sessions. Okay, thank you.
Thank you, Dr. Warren, for a very informative seminar. We can take some questions, even though we are short of time. Would anyone like to ask any question to our presenter?
Yes, may I ask a question, Dr. Bharat?
Yeah, sure, please.
Thank you for your great presentation, Dr. Warren. And I have a question about pathologic reports for oral squamous cell carcinoma. Is it important for you to have a depth of invasion of oral squamous cell carcinoma in pathologic reports? Because oral surgeons in Iran are very curious about depth of invasion, and they just know and they ask us to depth of invasion of oral squamous carcinoma in pathologic report meticulously. And is it important for you for your treatment plan?
Yes, because of the definition is one part of the T staging in the TNN staging in the HACC. Because of the depth of invasion can tell the patients about the prognosis of the patients. This one is one part of important information for the patients. But for the treatment planning, maybe not so much for me, for me and my centers. Okay, because of the most of my treatment planning for the surgery, maybe I have to evaluate it truly, not only for the depth of invasion from the pathological results, but all of the physical examination and the imaging.
Okay, great. Thank you.
Thank you for your question, Dr. Shakib. Would anyone like to ask any question from our speaker? No. So, Dr. Mohammad Kami, I request you to present our next presenter, please.
Thank you.
Okay, thank you. Thank you, Dr. Warin, for your presentation, nice presentation. And now it's time for the lecture of Dr. Sara Pourshahidi. She is Associate Professor in Oral and Maxillofacial Medicine Department in the School of Dentistry, Tehran University of Medical Sciences. At the same time, she is Vice Dean for Educational Affairs of the school, and her field of activity and interest is laser therapy. So we are ready, Dr. Pourshahidi, to hear to your presentation.
Alrighty, okay. Dear audience, I'm honored to be here with you and have your attention in these moments on my topic, different aspects of laser application in pre-malignant lesions. What we're going to talk about in these moments are pre-malignant lesions. What are they? We will have a glance at that. Pre-malignant lesions, lasers, and the effect of that, and the benefit of using that in pre-malignant lesions. And finally, I will address some concerns of using lasers in these kinds of lesions. What are oral pre-malignant lesions? These lesions have more potency and more tendency to malignant transformation than normal tissue. As a clinician, we should pay more attention to their follow-up after managing the signs and symptoms, because exact sessions of follow-up is very important in early diagnosis of any changes and any changes of transformation to carcinomatous changes like that. Leukoplakia, erythroplakia, and lichen planus are the examples. What's laser? Laser is abbreviation of light amplification by stimulated emission of radiation. And in this topic, waves in monochrome, in coherent, in collimated mode are irradiated to tissue and can have two major effects, like high power and low level effect on the tissue. I'm here talking about the high power effect of laser on soft tissue. Now I'm talking about that. Lasers can be used instead of blade here. We can cut with the laser, and we can benefit from its properties, something like less bleeding, somehow less pain, and something like that we're going to talk about in later, in further slides. And on the other hand, we can use laser to change specific lesions to non-specific ulcer. What does that mean? Something like the effect that we can have from using cryosurgery. This changing effect they can help us to change some chronic lesions with specific underlying pathologic phenomenon to a specific ulcer non-specific ulcer that we expect to be healed maximum in 2 weeks. But what's low-level application of laser? It's a non-thermal application. The patient will not feel anything, no thermal effect. The patient cannot even realize that you're applying low-level laser. And as a clinician, we cannot see any growth clinical change using and applying low-level laser, but we encounter a magic effect that is a photobiomodulation. Reported effects of low-level laser is, for example, avoiding apoptosis, increasing cellular proliferation, increasing ATP uptake, increasing cell membrane permeability to calcium, regulation of growth factor and cytokines, stimulation of cell differentiation and proliferation, synthesis and remodeling of collagen, increased tensile strength, angiogenesis and pain relief. And main clinical effects of low-level laser are anti-edematous effect. They have wound healing effect, analgesic response, and angiogenic effect. I want to talk about the inflammation and the effect of low-level laser on inflammation. It has a dual effect on inflammation according to the level of inflammation. Acute inflammation using low-level laser therapy will be accelerated. The good example for that is immediate pain relief of recurrent osteosarcoma after applying low-level laser. It's the example of acceleration of acute inflammation in that lesion. But for chronic inflammation, these kinds of lasers have anti-inflammatory effect that, the good example here is, for example, for arthritis, using low-level laser, we reduce the pain, the inflammation-related pain of arthritis. And let's talk about leukoplakia. You know, we all know leukoplakia and deal with that. But what's the benefit of applying laser for these kinds of lesions? Better control of bleeding is one of the advantages. Mostly we don't need to suture, and in some cases that access to the lesion is not easy, using laser here, it can be very useful. Of course, now I'm talking about high power application. When I'm talking about cutting, doing something like that. Precise tissue removal is another benefit, less morbidity and complication, and excellent healing. These are advantages that encourage us to use laser for these kinds of patients. As you can see in this review article and in this table, you can see that several kinds of lasers can be used for these kinds of patients. CO2 laser is mostly used, Nd:YAG, KTP, they are named here, but we know that we can use erbium YAG, we can use erbium chromium YSTG, and also even dyed lasers can be used to manage leukoplakia. And having a glance on the results of several studies, we can see that the recurrence rate and malignant transformation rate is not very different in these cases that underwent to laser surgery with the conventional surgical process. So it seems to be safe here. Lasers can be used for excisional application when a localized leukoplakia is there, and when these kinds of leukoplakia is on non-keratinized epithelium, some kinds of lasers such as CO2, KTP, and dyed lasers can be used in on-contact mode, and we don't need suture. I mentioned it. But about another kind of application of laser here, is vaporizing, and what where we use it when a lesion is comprehensive, when it is in attached gingiva and or on hard palate. They're very hard to be excision. So we use the vaporizing mode of laser, and different kinds of laser can be used to here again. This is an example of laser surgery for leukoplakia. And this is another example for that. Okay, let's talk about oral lichen planus, a prevalent, somehow we see more because we are the center of reference, but really they are prevalent, and they're common mucocutaneous disease that are classified in pre-malignant classification. Okay. For oral lichen planus, we know that they are not curable, and just we manage that. We use several things, for example, several remedies for that. For example, we use corticosteroids that have their own side effect. And in several literature, we can see that lasers have been recommended for even instead of corticosteroids, and mostly in combination with that to eradicate and to eliminate the signs and symptoms of oral lichen planus. Lasers can be used in high power mode, as I said, in replacing specific ulcer of oral lichen planus to a non-specific ulcer. And this is the type of management, but also it can be used in low-level therapy application. And in this mode, it affects the inflammatory process. It has two kinds of effects, primary and secondary effect. Vasodilation, lymphatic drainage, cellular activity and metabolism, blood flow, and activation of fibroblasts and neutrophils are the effect of are the primary effect of laser, and the effect on prostaglandin, immunoglobulins, lymphocytes, and beta endorphin are the secondary effect. Lots of things can be said in this on this topic, but we don't have time. Just we name that. But something that I should say is that these effects are localized again, and again I emphasize on that we don't cure oral lichen planus with lasers. Just we can have localized effects. But which kind of lasers can we use for these kind of management? CO2 lasers, in these slides, you can see that application of CO2 laser is effective in management of oral lichen planus lesions. In focus mode, we can vaporize the lesion, and in defocus mode, we can have the hemostasis, and we can encounter the re-epithelialization within 30 weeks, and after that, we can the symptoms are disappeared. And in this study that I am talking here and showing the pictures of that to you, after 1 year follow-up, there was no recurrence for the patient. This is the first ulcer, and this is after healing. So another kind of lasers are excimer lasers. I ask you to pay attention to these slides and help me if you have any idea about this kind of laser application for oral lichen planus. Let me talk more about this. This was an article that talked about non-patients that was irradiated. Their oral lichen planus lesion was irradiated by excimer laser. I attract your attention to the wavelength of excimer, and the author reported that the treatment had been painless and 5 patients had experienced excellent improvement, and just one of them was poor responder, and he or she had chronic active hepatitis C infection in that area. And they reported overall improvement, statistically significant here, but and the conclusion was they concluded that low dose treatment with excimer laser is very effective in treating symptomatic and especially erosive lichen planus. But my question is what to be concerned about? Do you think that it is safe? Okay, no idea, I will go on. If you see this spectrum of wavelength, we can see that the excimer laser is classified as UV laser and is in UV spectrum. And applying UV spectrum laser into cell is not safe because it can penetrate the nucleus and it can make mutation. So especially this is applied to a pre-malignant lesion that it is not, that has the tendency to go toward malignant changes. So in my opinion, it is not safe. It should not be done, and these kinds of articles should be thought critically on that. Okay, but another type of lasers that are more prevalent, cheap lasers, and they can be found in lots of dental and oral medicine clinics, are dyed lasers. Dyed lasers can be used to treat and management of lichen planus, even in high power and low level mode. And they seem to be effective. There are lots of reports on that. And as you see, they are dyed lasers mostly are in visible and near infrared wavelength, and they're safe from this aspect. And so everything is okay and this is effective, but again, I ask a question about your concern about that. Do you think that it's safe? Do you think that giving more energy, increasing ATP level for a cell that has tendency to bad changes, to malignant changes, is fine? Is it okay or not? And this is not just my question. This is the controversy, and this is a big question in area of lasers and pre-malignant lesions. So we should consider the effect of lasers on dysplasia to predict the effect of laser and its effect on transformation to malignancies. So the question is that, can photobiomodulation push the pre-malignant lesion toward more severe dysplasia or malignant changes, or is it safe to use that, and is it safe to manage these kinds of lesions with less corticosteroids, which have its own side effect? This question is this: how does dysplasia affected by laser, by low-level laser? For that, for responding to this question, we designed a study. At first step, we induced dysplasia in animal study, in animal model. We had to do it in animal model because the ethical problem of doing clinical trial in this topic, and also we could not use as you know, cell line, because dysplasia is arrangement of cells in tissue and not in cell level. So we had to do an animal study, but having dysplasia in animal model is very difficult because it's a borderline. In one side is normal, and after just this border, this narrow border, it goes onto SCC, and so it was a great thing, and we found a way to make dysplasia in animal model. This is our article that we introduced the way and how we made dysplasia. We stopped the changes on dysplasia in animal, in mucosal patch of hamsters. We painted mucosal patch of hamsters with a DMBA and stopped in dysplasia. And after that, we irradiated two kinds of laser to these dysplastic lesions. One in the red spectrum, a visual spectrum, and one in near infrared spectrum. We found laser to have no effect on severity of dysplasia, so we found it safe. But in this study that we published here, is the changes was observed in cellular, cellular changes was observed, and we are continuing these studies with the next step, which will be published in near future. It is on the epigenetic effect of lasers on these kinds of dysplasia. Just I can say that overall it was safe too. The other question is how SCC cells can be affected by low-level lasers. The question should be this, that do we really apply low-level laser to SCC? Directly, no, because we don't know why should we apply this kind of laser, soft laser, to SCC cells. Directly, no, but we should know that low-level laser is one recommended remedy for oral mucositis after radiotherapy and chemotherapy of patients undergoing to surgery with because of SCC. And so we should consider that maybe some squamous cell carcinomas cell have been remained, or even we consider the field cancerization. So if we apply and if we benefit low-level laser to manage oral mucositis after these kinds of treatments, we should pay attention to these kind of effect and the effect of low-level laser on squamous cell carcinomatous cells. So we designed the study again on this, and we studied on the effect of low-level laser on proliferation and invasion of cancer cells. We applied again two kinds of lasers, one again in visible spectrum and another in near infrared. More prevalent kinds of laser applied here in these patients, and we found that laser irradiation was significantly inhibitory. They had inhibitory effect on cell proliferation. We don't emphasize now on the inhibitory effect, just we can say, okay, so if we use this, it can be safe. According to these studies, of course, we published this again, and we are continuing our studies on this line. I'm going to conclude, have a conclusion here. According to our studies and according to the literature now, we can say that today, we can say that lasers seems to be safe for these kinds of lesions, and we can benefit from the devices for these patients, but we know that still we need more investigations and we have a long way to go on this topic. Thank you for your attention and time, and I'm here if any questions is there.
Thank you very much, Dr. Pourshahidi, for your impressive presentation. This is another aspect of oral cancer treatment, laser therapy, that you are somehow expert in. And I'm sure that all the audiences learned something about these techniques and these methods of treatment of oral cancer. So now we have time for question and answer. Maybe one or two questions can be answered. Please, if anyone have question.
Okay.
Just raise your hand and. Okay, dear Professor Vipavi, please. We are ready to hear your question.
Hello, thank you very much for the excellent presentation. I just wonder if anyone look for a recurrent rate after laser therapy on oral cancer. Is it high or is okay with the recurrent rate?
Okay, an oral cancer, you say that, yes?
Yes.
And there are some articles on that, and I told that there is no conclusion that it has changed. For example, we can say that which kind of laser application do you think about? In high power application, it's when we use. Let me explain what I understood about the question and then answer that. What I got is that the question is, do we use lasers for oral cancer, and does it have effects on the recurrence rate? Yes. Is it okay?
Yeah, I just wonder how high is the recurrent rate after laser therapy, because when we compare to a conventional conventional surgery compared to laser therapy, is there any difference regarding the recurrent rate?
Yes, I couldn't find any difference on this topic, really, and I'm thinking on that because really with when using lasers mostly is applied when high power lasers mostly can be applied when they are doing the first biopsy for that, I think, and I have not seen any report on the growth surgery with lasers, but as the answer of your questions, Professor, I have not found any difference on this recurrence rate in the literature since now. Me don't have encountered the difference here.
Okay, thank you.
Okay, thank you. So, and the Professor Shakib.
Okay, thank you for your great presentation, Dr. Sara. I know you are Vice Dean for Educational Affairs of undergraduate students. Therefore, regarding your position in dental school, I have a question. What's your idea about to include teaching or training students, undergraduate students, about laser therapy? Is laser therapy or an introduction to laser is involved in curriculum of undergraduate students, and what is your experience about it?
Dear Dr. Shakib, we in our school have some optional courses in our curriculum that we mentioned laser treatment or an introduction to laser in one of them. So our students can, if they want, can choose and learn more about lasers in a different course. But in a different parts of our clinical training, our professors will teach and will talk about laser application in different treatments of dental practice and of course soft tissue practice. But beside of that, we have another course that I mentioned. It's optional, and we can introduce more lasers to students that they want.
Okay, great.
What
Thank you. Okay, yes. I will be very grateful if Dr. Bharat or other colleagues from Thammasat University to share their experiences about training undergraduate or postgraduate students about laser therapy or introduction to laser treatment or something like this, if it is included in their curriculum or not. Thank you for your great presentation.
Thank you.
Okay, thank you very much. So maybe if there is anything, Dr. Bharat, you want to talk about regarding Dr. Shakib's question? Because we have another question. Yes.
Yes. I think Dr. Waring would be the one who can answer this question. Dr. Waring, would you like to share about the undergrad curriculum?
Okay, can you hear me?
Yes.
Okay, for the undergraduate curriculum of our countries, maybe the laser therapy is currently not included in the curriculum. The laser therapy, in my experience, I have a little experience for using the laser for the pre-malignant lesions in my residency trainings, maybe in Thailand. We start for the learn how to use laser in just some centers in the postgraduate in the oral surgeries divisions like this. But for me, it's a very interesting technology for use for the treatments, because the lasers, many cases can help the surgeries more easier, because the laser can stop the bleeding and it can have the clean wound more than the conventional surgery. So I think in the future, using the lasers regularly in the general dentist for just the minor surgery is, I think, is a very interesting.
Thank you very much.
Thank you, Dr. Waring.
Thank you very much. I think Dr. Piafong wanted to say something. Do you want to speak, Dr. Piafong? Yes.
Yeah, I
yes.
just want, actually, Dr. Waring already said everything. Okay. I think the limitation in Thailand is that we don't have many specialists in laser training. I mean, come up of my head, there is only one dentist that I know specialized, that she is specialized in laser treatment. So in many dental school, we only teach them just only basic. Even for us, we haven't seen the real of using laser. But as Dr. Waring said, I think that it has a role in the future if the technology become like more available, or we have more people who are specialized in the field.
Okay, thank you very much. And we have just one other question by Professor Mandana. So please, we hear your question.
Hello, thank you so much, Dr. Sara. That was very, very interesting. I have only one query actually. Throughout and in the beginning, it has always been that as far as oral cancer goes or oral pre-cancer goes, we have always focused on the proliferation rate. But some years back, we found that the rate of apoptosis is equally important, and it is reduced in all these regions. Now, in the beginning of your lecture, in one of the slides, you showed that the apoptosis rate is reduced. Now, while that may be a good thing for many other kinds of lesions, it's not a good thing in pre-cancer and oral cancer. So have you by any chance checked to see if there is a change or what is the rates of apoptosis in these lesions after you treat them?
Thank you, dear doctor. As I mentioned, because of these reasons, there is a controversy on application of lasers on pre-malignant lesions, and that's why my lecture's topic is on that, because lots of before these huge amount of study, most of the scientists thought that we should not apply laser for pre-malignant lesions because of these reasons. And we are doing, and not we, just we, but all investigators on these topic are studying, and the conclusion now, till today, it can be said that it is not as harmful as it can be thought. And we can say that now it can be safe, but we should always compare the cost and benefits. And if we have adopted using that on specific patients, so we can choose another way. But somehow, in some circumstances, this kind of therapy and treatment can be the only way that can be chosen. So we can consider this opportunity for these patients. We say that we can consider. But as you mentioned correctly, we should compare cost and benefits and also the literature till now. Thank you.
Thank you very much. We are somehow behind our schedule, but let us have the last question by Professor Mrna. So please.
Hello. So I like to just add on the name of the professor that she's one of the key person in laser treatment in Thailand. Her name is Prathan, and she's actually our part-time clinician who oversees our dental students, both master degree and undergrad. So if we are thinking about collaboration, I am sure that Dr. Prathan would be another key person for you to work with regarding like laser therapy. And she's in the Department of Oral Medicine.
Very good. Very good.
Yes, thank you.
Okay, thank you very much. So, okay, Dr. Bharat, we can go to the next presenter.
Thank you, Dr. Mohammad Khami. Our next presenter is Dr. Wari, but he would be speaking on a different topic. Dr. Wari, please, yes, the stage is yours.
Okay, all right, I'll share my screen. Okay, all right. Can you see it?
Yes, we can see it.
Okay, thank you. Okay, thank you. Okay, I have another session. For this test session is about my research field. That's about the artificial intelligence, and I would like to share about my research and some of literature reviews from the use of the artificial intelligence in the head and neck cancers. Okay, for the first one, I would like to share or introduce the terminology of the artificial intelligence. Normally, the artificial intelligence means the computers that mimic the human intelligence. It starts for I think more than 60 years ago. That for the starting is something like the novels or something like that. Maybe it cannot make it come true, but for now you can see that we can use for the computer for things like the humans. Is the mean of the artificial intelligence that we create for the artificial thinking from the computer to mix the humans. And the next one is the machine learning. I think I would like to share what this is. Machine learning is something like a subset of the artificial intelligence. Is something like the ability to learn without the program. It's something like the after we input for the information to the program or the machine learning, it can have the algorithm to further learning about this. For example, I think something like the Netflix. Do you know the Netflix? It's something like when you searching for the movie. When you search for the first movie, after that, the program can show you that the some gen or something the other movies that are so similar to the first one that you search. It's something like the machine learning program that recognize that what is your interest for, something like this. Or the Google, for the Google searching program, something like when you search for example, when you search for the oral cancer in the Google, maybe many persons search for oral cancer and search for oral squamous cell carcinoma, and so after you search using for the oral cancer, maybe it can show for the oral squamous cell carcinoma. It's something like the program that is something one kind of the machine learning that the computer program can learn and remember what the user used for. This one is the one of the artificial intelligence. And the next one is I think it's current technology in the most advanced in the artificial intelligence is called deep learning. The deep learning is something like is also the subset of the machine learning, but it has a more complex algorithm that we use for the mimics for the human neurons. Or maybe you can find the other words of the deep learning is the neural network or convolutional neural network that is mimics our brains. This one is one kind of the most advanced in the artificial intelligence. How can it work? I show some of the algorithms. For example, algorithm that as you see in our brains, our neurons has the many neurons and has the synapse of many neurons to work to make our brain work in thinking or making or doing anything. And for now in the computer science, we interest that if we can mimic the human neurons to use for the program, for the algorithm, we can further create for the more advanced in the artificial intelligence. That is something like that you see in the hidden layer or input of my algorithm of the picture in the upper left side. This one is for example, is how the deep learning thinking, how the deep learning is making to mimic our neurons. Okay. I would like to share one of the terminology that you need to know before I start for the deep artificial intelligence in the head and neck cancer. The first one is currently we use the deep learning in the term of one of the deep learning is object recognition. This is a program deep learning that use the program to recognize the object. For the recognized objects, currently it have the three terms. The first one is the classification. The second one is the object detection. The third one is the segmentation. I will show you what this mean for these terms. For the first one is the classification. I would like to share you a very simple picture. That is the picture of the dental implants and normal tooth. That is the first one. The classification is mean that the program can classify what this is. Is this A or is B? On this picture is classified. The program can classify that if we input for this picture, what is it? Dental implants and what is the normal tools, something like this is the classifications. For the next one, it's object detection. Object detection is how we after we input for the image. Okay, this is the same image. And after that, if we input that, we want the deep learning to recognize the object detection, how to detect the dental implants. This the program can detect that this is the dental implants and this is the normal tooth, something like this. This is the program, the deep learning algorithm that can use it in the term as the object detection. That's how to detect what it is. This one. And the last one is the segmentation. The segmentation is, I think, is most difficult to understand. It's something like the algorithm had ability to segment the image. That's we input for the. I would like to have the boundary of the dental implants. The segmentation is something like it has the detail and information of dental implants, and the program can segment the dental implant from the image like this. Is something like the segmentation. This T term is the term of the object recognition, which is the part of the deep learning. Deep learning not only have the object detection, you can or object recognition, something like maybe the data size or something like that is also some part of the deep learning. But today I would like to share only for the object recognition, something like this. Currently, the deep learning have many roles in the medicines, especially in the medical field. I would like to share the first one is the use in the clinical picture. In this picture is the skin lesion with the melanoma. The second one is using in the X-ray. The most common X-ray that's used currently use the deep learning or artificial intelligence is the chest X-ray. This one is the research to analyze the lung lesions. As you see, that the AI can recognize that after input for the image of the chest X-ray, it can detect the pneumonia in the right upper lobe of the chest X-ray. The last one, but has the many publications on the is the historical analysis of the something like this picture is using the deep learning for analyze the pathological specimen to have is it a malignancy lesion or not, something like this. This one is the role of the artificial intelligence in everyday life. This is something of the research that can, I think, in the future can use for the clinical practice for sure. Okay, let me start for deep learning in the head and neck cancers. I would like to share, and for example, of some regions. The first one I would like to share is there is a study in using for the deep learning for the detect of the esophagus cancers, esophageal cancer. The next one is the using for the laryngeal lesions in the laryngeal mass, or the last one is I would like to share my research about the use the deep learning in the oral cancers. For the first one, the publication is about the using the deep learning or for the esophageal cancer detection. How to about this method is I have to explain for some basics of the AI. The AI research. The first one we need is the data set of the image. Data set is the picture of the lesions with the lesion or without the lesions to input for the algorithm to learn. And how the algorithm learn it, we need the expert or specialized in this area to identify. It's called annotations, as you see in the picture, to annotation what or where of the lesions and what is the lesions that identifies is may call in the artificial intelligence or AI term as the ground truth. Is something like what it is in this picture is the using the expert to annotate or labeling the lesions, which is the esophageal adenocarcinoma, as you see in the picture, and then we input the image with the lesions and without the lesions to use the algorithm. In this research, this use algorithm is called, the first one is the RCNN, the second one fast RCNN, and the third one faster RCNN. Is something like the difference version of the RCNN, and the faster RCNN is one of the most novel of the RCNN series. And the next one is the SST. All of this is the name of the algorithms that use of that there inserts the picture with and without the lesions. After that, the result of this research, as you see, as I explained earlier, that's the cartoon. Cartoon is mean the lesions that are identified by the expert. In the clinical picture, is maybe this one esophagus is maybe had to identify by the general surgeon. And if it's the if it's the X-ray image, maybe need to identify by the radiologist, something like this. And this one is the using for deep learning to what is the performance to detect the lesions. It found that very interesting. The APR or precision rate recall something sensitivity specificity of F measure is something like the parameter to evaluate the performance of the model. But okay, I will summarize for you is something like accuracies. Okay, in this research I can summarize that something like that. The faster RCNNs and SSD algorithm can analyze the esophageal lesions with the accuracy around 80 to 90%. It's very, I think, is the really great performance for the AI to use for the detect of the esophagus lesion. Because if we in the future, if we can include it in for the practice, as we know, this view of the esophagus is normally from the esophagoscope of the general surgeon that had to examine the esophagus lesion. If we had the AI to include in this practice, maybe the AI can help the clinician to think and diagnosis of the lesions earlier. And it's something like imaging program. So the imaging program maybe it can help the humans for the medical practice for sure. Second research I would like to share is the research about how what is analyzed in the laryngoscope for analyze the laryngeal mass. As we know, one of the ENTs cancers that are most commonly found is the laryngeal cancers. Laryngeal cancer, as we've seen, as using the laryngoscopic imaging is something like in this research, using the deep convolutional neural networks, which is deep learning to analyze the lesions, and the result is using for classifications of the performance of this algorithm. The first one is something like analyze two class. Two class is mean that analyze how to classify that program can tell what is the lesion or the normal tissue, and it found that the sensitivity and specificity is, I think, is great. 70% to the 90% is great. It's something like, but okay, it's great. And the fourth class is something like device that how the computer age this algorithm can tell what is this, what is this cancer or just a benign or the normal tissue, something like this. Is something like in the AI term is called the multiple classifications, something like this, some multi-classification, something like this. And in this research, the most interesting part is they compare with the expert, and it found that the performance of their deep learning algorithm is comparable to the expert with 10 to 20 years work experience. Is that because of it, if we teach the algorithm and if we can make the effective model, maybe in the future this one can help the clinicians to solve many medical problems. And for now, okay, for the next one is for using for the AI for the oral cancer screening. Okay, I want to show you this picture, this one, and this one. As you see, if it's this one is the OSCC, the oral cancer. Okay, with our eyes, we can see it clearly that is not normal in the mouth. Is there something like the, maybe if the clinician has some experience for the oral cancer, maybe they can differentiate the diagnosis as the oral cancer and refer for it, because of this lesion is the obviously seen by our eyes. But the next one, if you see this picture, or this one, or this one, this is something like we found in our daily practice, that the patients maybe come to our dental clinics and have a dental filling, something like this, but have the lesion, but they have no any symptom. And mostly, if the lesions represents as the picture, as this or picture, maybe the clinicians mostly misdiagnosis and misdetection it for sure, for my experiences, especially for the unexperienced clinicians. But as you see, this one is the, or this is the OPMD lesion or pre-malignancy lesions, that if we miss to recognize it can lead the patients to, and it can lead these lesions to transform into the oral cancer. So this is the problem. This is the research of my research problem. That is, if we can use the computer vision technology, for example, the artificial intelligence, can we use it to help the clinician to detect it? Because in our practice, just our eyes, maybe it can mislead or misrecognize it. Okay, for my research is using the deep learning algorithm, which is the subset of the in artificial intelligence used for the oral cancer detection. Okay, I have my publications on using the deep learning algorithm for the oral cancer, which is OSCC and the OPMD public. You can read for the full detail in my publication or my original article. But I want to show you for the first one is the deep learning role in the oral cancer detection. In my experiments, I input the data of the oral image, oral image. The first one is no lesion, it's a normal oral image. The second one is the OSCC image. All of my data sets OSCC image has the pathological result as the gold standard for the diagnosis of the OSCC. And for the AI research, the first thing after we input for the image, we need the expert to annotations or label where is the lesions, as you see in this one. And after that, I using the algorithm to classification and detection it. I would like to expand about the AI research. That's the first one, how we make the algorithm or how we make the model. We can divide after, for example, I input all the image for 700 image. After that, we have to divide it into the 3 groups. The first one is we call the training. The second one is validation. The last one is the testing. The training is mean using all of the image to develop the model. The model from the algorithm that we is interested and we select to use this. The second one is validation. It means to tuning the model. To tuning that model is okay for using in this type of data set. And the last one, testing, is to test the performance of the model. And the most important is all of the 3 groups need to be the different image or different data set. In our results, the first one, the classification performance, we select the denseness 1 to 1. It found that it can classify the oral cancers with the AUC. AUC is the one parameter I can tell that is something like accuracy or performance of the model. It can tell the lesions, the oral cancer lesion apart from the normal image with the accuracy, with the performance around 99%. And the second one is the detection performance. We're using the faster RCNN, and it shows that the accuracy or the performance of our algorithm is around 80%. I think it's something like that. This algorithm, this deep learning has an potential for using for detect of the oral cancer lesions in our oral cavities. The next one, I also interested. Okay, as we seen, the oral cancer is obviously seen by our eyes, but for the lesion that may be easily to misrecognize, which is the pre-malignancy or OPMDs, how is the deep learning algorithm can detect it and can help the clinician for it? Okay, for the experiment, it's also the same, using for the no lesion and OPMD lesions. OPMD lesion including or erythroplakia, leukoplakia, or recurrent image. And we annotation where is the lesion, and using for this time, I use more than one algorithm for the classification and detection because of the lesions. Normally, it can mislead by the clinician. So I would like to know, is the algorithm okay for detect it? And then I develop for the model and test it for performance. Interestingly, the classification for performance is also high for both of the denseness 1-to-1 or lessness 5-0 is a AUC of the 95%. And the detections, as we've seen that there's something like the OPMD lesions may be in the limits of the lateral tongue. As you see, the red lesions may be normally from our eyes, maybe if the unexperienced clinician may be easily to mislead it, but the algorithm can detect it. But some of algorithm may be not appropriate for using for the detection of the OPMD lesion, as seen in the Euro before. That accuracy is not so good, but the faster RCNN is okay. Another one is in this research, I also compare the classification performance of the AI to the expert, and I found that is comparable to the expert level, the board certified oral and maxillofacial surgeons. That's the performance of the classification performance of the denseness 1-to-1 and lessness 5-0 is, I think, is really great. So in the oral cancer screenings, as we know, the oral cancer, the prognosis is that for now, it depends on the staging of the oral cancer. So if we can screen it in the early stage, and using the technology such as the AI technology can detect and help clinicians to detect it earlier and make the patient's treatments early, I think it can help to improve the prognosis and increase the 5-year survival rate for the patient. So I expected that the deep learning algorithm of my research can develop into the mobile applications, which can use for the oral cancer screening in the future. That can help the clinicians, especially for the inexperienced clinician or general dentist, to detect of these abnormal oral lesions. Thank you.
Thank you, Dr. Waring, for your very informative seminar. Really appreciate for your time and efforts. The platform is open for questions and answers. Would anyone like to ask any question to our presenter? Yes. We have Dr. Pia Pong.
Hi, Dr. Waring, thank you very much for sharing your research with us. My question is that the image that you use for analyze, is the quality of image also affect the result? Can we use the, let's say, photo taken by a smartphone or mobile phone? And also sometimes it's quite difficult to take a photo in the mouth. What is it possible to use like flash from mobile phone if we don't have like a well-equipped camera?
Yes, in the AI research, if we have the more different data set or more different some kind of the data, as Dr. Piyapong asked me, is the mobile photos or the photo from the camera? Okay, we know that is that has the different quality of the image. And if we input for the AI research, if we input the different quality, the more different for quality, it can make the AI to learn the different one. And if we input the high quality and low quality, after that, if the more something like adequate data set, the AI can learn to analyze both of the picture from the low and high qualities of the image. So for the AI image, I think it's very interesting. We include all of type of the image qualities.
Professor Inglam, please go ahead with your question.
Okay, thank you. Dr. Kisasit, do you know Bayesian network? In the past, Professor Siriwan, my research, used Bayesian network to predict endodontic treatment outcome. What is the difference between Bayesian network, neural network, and deep learning?
Okay, it's an interesting question from Dr. Ingram. And that research is a very interesting research about using the Bayesian network. It's something like a different algorithm that has different role. The Bayesian network is commonly used to analyze the data to predict the prognosis, the prognosis from the data type of for the data, or the how can I say, the data set as the data, something of like the data. But for the deep learning, and in my research as the object definition, use the different data set as the image data set, something like that. And so for the conclusion is that used for different roles. Okay.
Yes, Dr. Bharat, I just then would like to add a little bit more from Dean Samrung, So as Dr. Warin presented in his research, so he used deep learning. That is one type of AI. But, there are other types of AI, like Dr. Ingram, Dr. Samrung said, for example, Bayesian network and other things. I think one of the algorithms that we are quite familiar is, for example, logistic regression. I think all of us are familiar with logistic regression. That is, very simple form of AI as well. So we take all the variables, for example, patient's variables as an input. That one we can consider it as what we call understandable AI. It is understandable AI. It means we understand how the algorithms work to come up with the output as a prediction. But for the deep learning that use images as the input, someone said that it is a black box AI. Black box means, we could not understand, how they come up with the good result like that, but it used image, vision technology, as Dr. Warin mentioned, but it has a lot of, potential in terms of prediction of the medical images. And as we see from the results from Dr. Warin, actually I am the co-author of the research project, we see the potential that it can predict, the malignant or the OPMD accurately, comparing to the expert detection as well. And this is what we call it's like the it's like the preclinical and no, it's the basics research. It means we really need a randomized control trial or the community application so that we can compare, the results of the system, of the applications, and also the expert or the general dentist prediction as well. So that is our future work. That is the idea that we can also collaborate with the Iran or Tehran University as well for this direction. Thank you.
Thank you, Professor Siriwan. Actually, this is what I was going to ask you, that are you organizing any workshops for undergrads or postgrads or even the staff who are interested to know a bit about AI, use of AI?
Yeah, I think that would be possible, that we can do the workshop so that we can educate, for example, as an introduction using AI in dentistry. I think that is possible as well. Yeah.
Thank you. Thank you, Dr. Siriwan. Thank you, Professor Inglam, for your questions. I request Dr. Mohammad Khami to please present the next speaker.
Okay, thank you very much, Professor Warin, for your impressive presentation. So the field of AI is progressing day by day, and we are in the age of information. So I think we will hear more about your research, I'm sure, in the future, successfulness in your research area. So, it's my pleasure to introduce my colleague, Dr. Atta Qarajei. Dr. Qarajei is Associate Professor in Oral and Maxillofacial Surgery Department of the School of Dentistry, Tehran, the University of Medical Sciences. He also works in Cancer Institute Hospital. He's the director of postgraduate students of Oral and Maxillofacial Surgery Department, and his field of interest and activity is oncosurgery. I think very interesting field. Dr. Qarajei, we are all ears to hear your presentation.
Hello, everybody. I'm so happy to be with here, and I'm very happy to share my experiences in this field with you. Topic of my presentation is reconstruction of mandible, mandibular defects with free flaps using virtual methods for improving results. My presentation is divided. I have divided my presentation in three main parts. At first part, I want to talk about basics of reconstruction of mandible, and then talk about the role of virtual surgery in this procedure. And at the final step, I will share some cases with you. Here is Imam Hospital Complex, one of the oldest hospital in Iran, and also one of the most important hospital. Our department, head and neck surgical oncology and reconstructive surgery, that's situated in this hospital. We have more than 1,500 head and neck surgery per year, and also more than 100 mandibular reconstruction with free flap in this department. As you know, defect of mandibular defects etiology could be ablative tumor surgery, traumatic injuries, congenital deformities, and also inflammation or radionecrosis. Most of our cases are ablative tumor surgery because of our center is referral for head and neck surgery. Goals of reconstruction is restore the lower third of the face, aesthetic and function, reestablishment of continuity, also reestablishment of normal position of condyle, normal occlusion relationship, also normal motion, normal oral competence, and also a lip and oral soft tissue sensation. In history of reconstruction of the mandible, we see many efforts for producing a new mandible with other bones. And this is very interesting and also very hard, because mandible is a three-dimensional form, and other bones in body that we could be able to harvest is not this shape. As you see, anatomy of mandible show us is unique bone in body that's very functional, and also it's important from aesthetic point of view in our body. And also many other muscular attachments that these muscles help to mandible to work efficiently. Defects of mandible classified in many types. One of the first classification belonged to Pavlov in 1974, and the last one belonged to Brown in 2016. Each of these classification approach to this problem from their own perspective. But most of them, and perhaps I can say all of them, focus on the amount of defect, site, and also the tissue that we need: bone defects, soft tissue defects, and also neurologic defects. These are different kind of classifications that we have now. All of these, the classification for facilitating the reconstruction problem for us. This is Brown classification. That's the last classification that I found in literature. One of the most important problems that we have is timing of reconstruction. Timing of reconstruction, mandibular reconstruction, widely debated in literature. Historically, proponents of a delayed or a staged approach because of recurrence that we have in these kind of surgeries. But nowadays, it's widely accepted that immediate reconstruction may be better if it's possible for us because of quality of life that we can dedicate to patient. Although in shorter life, but with high quality. Immediate reconstruction actually minimize the cascade of scar formation associated with initial injury, and also significantly improve quality of life. But in secondary reconstruction, although we have some problems according to scar and also many other problems in surgical technique, but some studies that showed that no increased rate of complications compared to primary reconstruction. We have three options for reconstruction of mandible: reconstruction plates, non-vascularized bone grafts, and also vascularized bone grafts. Reconstruction plates today we are using very rare because of high complication rate, plate exposure, low success rate, and also further complication rate in radiation therapy, in combination with radiation therapy. Reconstruction plates alone are prone to failure, and these days we have not used never. Non-vascularized bone grafts done only for small defects. It's not possible to using this kind of reconstruction for massive defects that we have in cancer surgery. But vascularized bone defects have many advantages. These are healing independent of compromised recipient bed. Also, for anterior mandibular defect, we have not any choice. Also, these kind of reconstruction have enough length to bridge the defect. Also, these kind of reconstruction have this opportunity that we reconstruct hard and soft tissue concomitant. And this is very, very important for cancer patients. We have many options, and all of them have some advantages and disadvantages. Radial osteocutaneous flap, that's we have not used many of this kind of flap. This is very narrow indication. And other flap is scapular osteocutaneous flap that need good experience. I myself have not experience in scapular osteocutaneous flap, but I know that some indication that's very good, especially in maxillary reconstruction. Deep circumflex iliac artery flap that we have very good experience in this flap, DCIA flap. DCIA flap has many advantages because it's corticocancellous bone, very good height, very good shape, and also pedicle is problem. One of the most important problem in this flap is shortness of pedicle and also the diameter of artery and vein. One of the other advantages is osteotomy that it be possible to do, and also according to the size and site of the defect, we should select the site for donor. And other disadvantages including poor color, bulky of soft tissue part of flap, and also skin paddle located on the external aspect of the iliac crest. And also some complication for after the surgery, including pain at the donor site, that some patients should complain about that. But fibular flap, vascularized bone flap, vascularized osteomyocutaneous flap, provide a long segment of bone up to 25 centimeter that tolerate multiple osteotomies without compromising its blood supply. That's a very important point and very important advantages for this flap. It's sizable and also lengthy pedicle on the pronal artery and size of this pedicle from point of length and also diameter of artery and vein is very, very good for anastomosis in neck, artery and veins. Also, it's possible that we harvested accompanying skin paddle that's vascularized by septocutaneous perforators from coronal artery, and also can be harvested with FHL, flexus hallucis longus, or soleus muscle to fill soft tissue defects. This flap has more than 90% success in experienced hand. And it's very important. This is a very important problem that we have with other flaps, but fibula flap is very, very good flap from standpoint of success rate. Some disadvantages about fibular flap: pain or ambulation and ankle instability could be one of the complication. Walking at high velocity or while performing complicated actions, it's more difficult for patient after surgery. Some patient with skin graft loss on donor site or dehiscence and perhaps compartment syndrome that has been reported for excessive tie primary closure of the donor site. Weakness of great toe flexion and other complication, and also limited height of fibula that introduced double barrier for technique for solving this problem. Algorithm for reconstruction of the mandible with any kind of free flap, including these steps: length and location of mandibular bone to be replaced, amount of intraoral lining and type of soft tissue structures that we need, recipient vessels availability, medical comorbidities, availability and appropriateness of the various pedicle and free flaps, and status of donor region. These are very important in any kind of reconstruction of mandible via free flap. Reconstruction on mandible in anterior part, any defects that include anterior mandible, it's better to reconstruct it with vascularized bone defects because of the form, and it's not possible to do with other options. For reconstruction of lateral mandible, we have other options, including DCIA, because of better height of DCIA than fibula, or using, as I said, is using double barrel technique for this kind of reconstruction. For condylar reconstruction, also we have some options, including titanium prosthesis or plating native condyle onto a vascularized bone flap. And in general, it's a very difficult part of reconstruction of mandible is condylar reconstruction. Osseointegrated implants have very, very important roles, especially nowadays in reconstruction of the mandible and function. We have one term, one jaw, one day, that's very interesting, but need very, very delicate treatment plan. And it's very, very hopeful future for us that it be possible to bring a patient to the operating room, and after resection, it's be possible to reconstruct all of the mandible and also dentition system in the same time. For virtual surgical planning that I want to emphasize in this presentation, we involve 4 distinct phases: planning, modeling, surgery, and evaluation. For planning, we use the high-resolution CT of head and neck and also donor site. Our cuts between 1 and 1.25. For modeling, we use CT scans that forwarded to desired company with a DICOM extension. In the company, 3D images of craniofacial and donor site constructed. After that, we held a meeting or web meeting with surgeon and other team. And also we talk about the resection size, resection limitation, and also other phases of our reconstruction and our problems that shared with each other. Stereolithography models are manufactured, osteotomy guides, both osteotomy guide for fibula and or iliac, and also guides for resection. All of them sent to operating room. All of them are sterilized. Access to in the surgery phase after the patient in operating room, access to recipient site is firstly done. Osteotomies according to cutting guides are done, and guides is placed on fibula fixed, and then resection, osteotomies at donor site with vascular pedicle being intact. As you see here, we have guides. And also, if it's possible, please, I want to show, okay. As you see here, at first step, fixed the guide on fibula flap. And then osteotomy has been done, as you see here, with preservation of pedicle behind the osteotomy site. As you see, we have this scene after osteotomy that we remove extra parts and after shape the mandible. As you see, we have a scene like this. It's very interesting because one long bone is now shaped to three-dimensional mandible form with enough muscle, enough skin. That's we have not any wasting time for. After surgery, we have evaluation of reconstructed patient, post-op CT scans, assessment of accuracy of planning, and also refinement of our technique as needed. In the last part of my presentation, I want to show some cases to you. I wanted to show one 45-year-old male that previously operated here 3 years ago with left hemimandibulectomy with suboptimal reconstruction. Pathology was oblongoblastoma, and reconstruction was done with fibula flap. As you see, this is before surgery. Patient referred to us with this view. This is modeling. We use double barrel here, as you see here, for increasing the height. These are the surgery scene and after fixing the free flap in the side. And this is the final view of the patient. Another case is 68-year-old white female, previously operated for lateral mandibular SCC 3 years ago. And now she has a complication of this defect in right mandible, in left mandible. We used mirroring for reconstruction of the exact location of mandible. These are after, before surgery, scene after surgery, and also, as you see, this is the same patient after 2 years. Case 3 is 65-year-old female that osteosarcoma. This is immediate reconstruction with fibula flap, as you see here. Case 4 is 30-year-old white female, SCC of left mandible, that reconstructed immediately with this flap. Case 5 is 25-year-old white female that she had amyloblastoma, and we had planned for reconstruction with DCIA, but using guided surgery. This is a very new technique for guided surgery, and only we have limited cases that introduced in literature. As you see here, we use the guided surgery and for forming the mandible from iliac crest. This is the same patient. This is the defect. This is the donor site DCIA that we harvested with pedicle after fixation and anastomosis. This is immediately after surgery, and this OPG 1 year after surgery. As you see here, it's very good form and function. And the last case is 25-year-old white female with multiple osteosarcoma recurrence. As you see here, this is the patient with very poor prognosis. We resect mandible angle to angle. This is the guided model, and we use 90% of fibula. This is the same patient after surgery and after 1 year after surgery, and this is also the same patient with mandible that constructed with fibula. Thank you for your attention, and this is my team in our university that we are working together. Thank you very much.
Thank you very much, Dr. Karaji, for your great presentation. It was very impressive. I would like to let know audience that it is my great honor to work with Dr. Karaji at Cancer Institute Hospital, and he is one of the most well-experienced surgeon at that center, and he has several cases monthly and reconstruct them. Let me ask you the first question from my side, that Professor, would you let me know how long we should wait from diagnosis of oral cancer, especially oral squamous cell carcinoma, for reconstruction? How long you would wait to evaluate recurrence or second primary tumor or something like this, and make decision for reconstruction of the region?
Thank you very much, Professor Shakib. That's an honor to me that working with you in our institute also. I want to, as I talk about in the presentation, the last trends was that secondary reconstruction, but today's trend is not. Today's trend is immediate reconstruction, although we know that the biology of tumors very different with each other, and perhaps we have not possibility to use this very large operation with good length of we have not, I want to say that perhaps we know that we perhaps lost our patients in very short time, but because of quality of life, we nowadays not secondary.
Okay, thank you very much. I think we have another question from Dr. Warin. Dr. Warin.
Okay. Well, it's a very interesting cases, very complex cases that you show us that. I would like to have the question. The first one is that what is your common free flap that you use at your for the mandibular reconstruction at your centers?
Thank you, Professor. In my center, we use free flap of fibula for 90% of cases, and for another 10% DCIA, and also for deep circumflex iliac artery flap, and for soft tissue defects we use ALT, and also rectus abdominis, and also radial forearm. These are our workhorse flaps, and also we use in many other cases pedicle flaps, submental, pectoralis major.
Oh, okay.
Okay, there was some problem in the connection, but now you are connected, Dr. Garajei. So, okay, thank you for your explanation. So, if there is any other question.
No, yes, I have a couple of questions. For the another question is after you had the bony reconstruction from the free flap, and did your center perform for the dental implants after this? If the patient had the dental implant placement after the surgery?
After surgery, we for dental treatment.
Dental implant placement.
Dental implant. We not use immediate dental implantation, but if it's possible for us after healing, we use dental implants, especially for benign tumors. Because in patients that we have radiation, it's not possible to do implantation because of osteoadenocrosis probability.
It's also the same problems for my centers, and also in the Thailand, that we want to reconstruction for the patients to be completely with the soft tissue and the bone that we dissection it. But the major problem, I think, in nowadays for the dental practice is for the denture, for the dental rehabilitation of the patients is very challenged, especially in the malignancy case that we need to send for the radiation therapies. Okay, thank you.
Thank you very much. So,
thank you.
Okay, is there any other question?
Dr. Etta, I would like to continue what Dr. Warin said. As a maxillofacial prosthodontist myself, I'm curious to know if you have any maxillofacial prosthodontist in your team. You showed a very nice picture of your team, and whether you are working with them to have surgical stands or surgical guides.
Yes, Professor, we have a prosthodontist colleague in our team because, as you know, many of these defects, especially maxillary parts, need these kind of obturators and also these kind of splints. And in some parts, we have maxillofacial prosthodontists that use for reconstruction of nose, and only we resect the tumor, and in some cases install implant, and after that refer to prosthodontic department for reconstruction of nose or eye prosthesis for them. Yes, we have a very great team in this regard. Yes.
Okay, thank you. So, is there any other question? Okay, so we can go to the next presentation. So please, Dr. Baharat, please introduce the next presenter.
Thank you, Dr. Mohammad Hamid. So I'm pleased to announce the next presenter, and I'm fortunate to be working with her, and she shares her passion for gerodontology with me. She finished her residency training in geriatric dentistry from University of British Columbia in Canada, and also PhD in dental science at same university. Currently, she is the Assistant Dean for Postgraduate Clinical Affairs at Faculty of Dentistry, and also the Program Director of MSc in Gerodontology. She is also a committee member of Thai Board of Dental Public Health and also Royal College of Dental Surgeons of Thailand. Her fields of research include geriatric dentistry and oral health care for special care needs. So please welcome our next speaker, Dr. Mrna.
Thank you. Could you please allow me to share the slides? I'm using another account. Could you let the host? Because now it said host disable participants screen sharing.
Dr. Mohammed,
you are co-host now. You can share your screen.
I'm using another one. Named Mrna.
You cannot share your screen?
I'm using my laptop, but
yes, okay.
My iPad, yeah.
Okay, just let us.
I'm sending the direct message to the host already.
I think it's okay now. Can you check that?
Okay.
Okay.
Okay, so hello, salam, and sawadee ka, everybody. The talk today is about the supportive care. The outline is as follows. So we'll begin with the definition, some general principle, and because we have only 30 minutes, so I'm going to give you some examples of the supportive care during the course of cancer experience of the patient before, during, and after the cancer treatment. Okay. So what is supportive care? Supportive care is both prevention and treatment of the cancer itself and also the side effects that are caused by the treatment of cancer. And it starts as early as possible. It involves the physiological effects and also psychological, social, and spiritual problems related to cancer and also the treatment of cancer. So that's a definition from the American National Cancer Institute. So the NHS of the UK adopted the definition written by the Multinational Association of Supportive Care in Cancer, and they emphasize that supportive care has to be across the continuum of the cancer experience, from diagnosis to the post treatment of care to secondary cancer prevention for the survivor and so the end of life, and supportive care and palliative care as well. The widely accepted international guideline is has been published by MAS, and you can see that there are several issues related to supportive care, whether it's a skin, like acute skin reaction, or neutropenia, or like nausea, vomiting, or palliative care. But the three groups that produced the guideline directly related to us, oral care study group, mucositis group, they issue the guideline both for oral mucositis and also GI mucositis. And the last group is the bone and musculoskeletal study group that they published a guideline relating to the jaw necrosis. And you can see that you can read the full guidelines. So you can see that the supportive care related to cancer is not only for head and neck cancer, but any cancer treatment that affect the mouth and the jaw as well. Whether it's a surgery directly to head and neck cancer, radiotherapy directly related to head and neck cancer, or chemotherapy of other parts of the body as well, including the stem cell transplantation as well. And also I like to suggests that we should not forget those people who refuse the treatment of cancer, that they also need supportive care as well. In Thailand, I think there are a lot of people who refuse the treatment of cancer, and we have to do the palliative care to the very end. Okay. So today I'm going to review guidelines and protocol of many hospitals, but mainly we would base on the international guideline written by MAS and ISO. There are a lot of different adverse effects from cancer itself, from the surgery, also from the treatment of radiation, stem cell transplantation, and chemotherapy. I cannot go through all the side effects or the adverse effects today, but the main principle when we read the guideline is there are recommendations and suggestions for and against such intervention. Recommendation would reflect a stronger level of evidence, while a suggestion for or against is based on a weaker evidence. Okay, and then we have to read carefully whether it's for before, during, or after the cancer treatment, and what would be the outcome? Is to prevent or to treat? For example, I'll give you the example of the latest guideline published last year of how to manage oral mucositis. Okay, like for Diflam or benzidamine, you can see that they recommend benzidamine to prevent OM in moderate dose of radiation, but only suggestion because of weaker level evidence of benzidamine to prevent OM in the combination of radiochemotherapy. Okay. And for chlorhexidine, they not only just suggest for, but they also suggest against the use of chlorhexidine for prevention of OM. Okay, but for treatment of OM, they said the evidence is inconclusive, so they cannot make any guidelines. This is the newest change in the guideline that they decided to go for a favor of the application of honey. It's a combined topical and systemic delivery of honey for prevention of OM, but it's only a suggestion, it's not a recommendation. So it poses a weaker level of evidence. The mechanism that they explain is that honey has the low level hydrogen peroxide generation and also non-hydrogen peroxide phytochemical mechanism of wound healing. And also honey itself has an antimicrobial property to prevent secondary infection. Okay. So because of low risk, they said, okay, they go for a favor to suggest but not recommend. The reason the level of evidence is not strong because they use, in many studies, they use different sorts of honey. So you can see that if you want to get the honey, you have to get, like you don't really know where to get the honey and which one has what property. And the study, even though they show the effect of improved outcome, but the study posed a small sample size and with different population differ, like some with the chemotherapy, some with the radiation therapy. Okay, so that's the guideline for professional use, right? But for patients, if you go into the website of the International Society of Oral Oncology, there are videos, like clips, and also patient education fact sheets translated into 19 languages, but no Thai and no Farsi. Like we cannot, we just have to read on the English version. And you can see the patient can read how to take care of the mouth during the active chemotherapy, or how to take care of a lot of structure in the body before, after, and during the head and neck radiation. Okay, so besides the mouth, what are the issues of supportive care? Eating and drinking, swallowing, skin care, tooth feeding, and care. So oral care is only part of the whole supportive care spectrum. Okay. I'm going to give you an example of the case we have at Thammasat. So this prescription written by physician, by oncologist, for the patient who had malignant neoplasm of tongue before, during, and after the treatment. So you can see that the physician, the oncologist also take care of the oral mucositis. They take care of the pain. So they prescribe lidocaine and tramadol. They also prescribe antifungal for thiamazone, like after the treatment that the patient developed fungal infection. So you can see that without a dentist, oral issue is addressed, but it's not comprehensive because they did not address the dental caries or the dry mouth issue. So we need a team to make a comprehensive care for the patient. Okay. Use a team approach, but we don't have to take like authorship that I have to take care of oral fungal. I have to take care of the pain in the mouth. I mean, if we're a team, as long as a patient has been taken care of comprehensively, whether it's an oncologist, whether it's a surgeon, whether it's a plastic surgeon, or whether it's a ENT or a skin doctor or a dentist, as long as the patient is fully taken care of in every aspect, then that's okay too. Okay. So that's what's happening in our hospital, because we do take care of cancer patients, but we are not really a cancer institute. Okay. But in some other hospital that like your hospital in Tehran, that you're a cancer institute, you have the whole team, and including like in Japan, they have a dental hygienist who actually take care of inpatients' oral hygiene as well. So it depends on the structure of the hospital that you are in, but as long as you have the whole team, it's okay. In Sweden, in Stockholm, they have a case conference every week. So every case goes through a case conference of treatment planning together between dental hygienist, dentist, oral maxillofacial surgeon, oncologist, general surgeon, like radiologist, like the full team with a case conference every Tuesday afternoon, get the treatment planning together. So that's like the ideal, the perfect team approach. Okay. The NHS in the UK also said that the supportive care team that work together also needs communication technology and also need the evidence-based protocol. So if in the institution we have these two in place, it will help the patient to start supportive care as early as possible. So from now, I'm going to just give some example of what are the supportive care strategies before, during, and after cancer treatment. So for before the cancer treatment, we all know that the patient has to go through oral examination and clear all the risks of dental diseases, right? But if possible, the baseline of the range motion and the baseline of saliva should be measured, should be assessed as well. And you can do the custom fluoride trace, right, even before the treatment of the cancer itself, to get prepared. And for the MAS guideline, they suggest that implementation of multi-agent oral care protocol is needed. So that include daily brushing, interdental cleaning, rinsing, and oral moisturizing. So I like to emphasize here that for cancer cases, try to avoid Vaseline or petroleum jelly and glycerin product, and try to use the water-based or lanolin instead. Okay. So you can see you can use like KY, which is very common to find, and lanolin, which is in Thailand, we can find it online or in the department store, but not as common as KY, where we can find in convenience store as well. Okay. So the reason to avoid the petroleum jelly is because of, first is the allergy, and second is it can turn into the exogenous typhoid pneumonitis in patients who are like really immunocompromised if they inhale or they aspirate the lipid part. Okay, so that's the before. Now the during cancer treatment, this is a newsletter from Johns Hopkins saying that with the proper oral care bundle, this program had led to a 50% reduction in mucosal barrier infection. The patient get discharged and go home 10 days earlier, and they don't have to go to ICU. So this is like a high impact from just proper oral care during the cancer treatment. Okay. So what do? They do just like standard oral care plus plus what? Plus suck ice chips. Okay, so the proper name is cryotherapy. So the latest guidelines said they recommend the use of 30 minutes of oral cryotherapy, use the ice to prevent oral mucositis in during the infusion of chemotherapy. So this is the final recommendation. But actually, if you look at the protocol of each hospital, there are many protocols. Some hospital let the patient sip or suck on the ice chip for 5 to 10 minutes before administering the chemotherapy, and then during for about 30 minutes and another half an hour after the administration. Okay? But the final recommendation is just during the infusion for 30 minutes. Okay? And also for the stem cell transplant as well. Okay, what else? For during the cancer therapy, we need to do pain management and management of dry mouth. And also if there are pain and bleeding, we have to take care of oral hygiene, maybe using some like super soft toothbrush. Okay? So I'm going to talk about pain management now. The previous guideline recommend like Benadryl, lidocaine solution or lidocaine viscous 1 or 2%. Later, the MAS guideline recommend 2% morphine mouthwash or 0.5 doxepin mouthwash, which is an antidepressant. Okay, but the latest recommendation is a suggestion of topical morphine 0.2% mouthwash in the patient or head and neck cancer patient treated with radiotherapy and chemotherapy. Okay, so it used to be 2%, but now it's 0.2%. For the rinse, they recommend a bland rinse. Okay. They said the data is limited, but the saline and sodium bicarbonate rinse are mild, and it should increase the oral clearance, which is helpful for maintaining oral hygiene and improve patient comfort. BC Cancer Agency state that you can also use magic mouthwash when the patient also have pain. However, there are limited evidence suggesting superior over bland rinse. Okay, so bland rinse is saline and sodium bicarbonate. How to mix it? Just the easiest recipe is to use 1 tablespoon of salt, 1 tablespoon of baking soda, and 1 liter of water. Okay, and you can use daily. Repeat the mixture every day. Okay, so throw away at the end of the day, and you will mix again in the morning. You can use every 2 hours, and be careful that sodium bicarbonate is baking soda, not baking powder. Baking soda is alkaline, and baking powder is acidic, so it's 2 different things. Okay. We need something similar to bicarbonate as a saliva. Okay. During the cancer treatment, avoid anything irritating. So anything with alcohol, anything with like strong essential oil for the mouthwash. Okay. And how often do we see the patient? They said assess oral mucositis at least once a week, or tell the patient to communicate like through telecare. And if the symptom worsen, then contact the dental team. The ultra soft toothbrush and foam brush. They said you can use ultra soft toothbrush or a soft toothbrush place it under hot water first to make it softer. And even though the patient feel discomfort, encourage them to brush anyway. And if the bleeding occurs, encourage gentle brushing, but still you have to brush. So they like said, you still have to brush, okay? And if you have oral infection, like fungal infection or viral infection, try to replace the toothbrush after the infection subsides. Right? So there are several types of toothbrush. This is not over the counter, but if our hospital, our department have all of this in place, so it would be easier for patient to access. Right? The sponge oral swab or foam brush is also included in the guideline in case that the patient bleeds a lot or has like really painful oral mucositis. But there's a caution of using this because of the dislodging or tearing of the foam heads from the stick. And in the UK, NHS has banned the use of the foam stick already. And for the NHS has launched a program called Mouth Care Matters for the inpatient admitted patient, and they changed to use the silicone-based oral swab like this instead of the foam sponge. And for the bleeding, so how much bleeding do we stop toothbrushing? So in the general rule is if it bleeds more than 2 minutes, so that's dangerous. The guidelines varies according to platelet count. The lowest I can find is the Cancer Care Ontario, the Canadian guidelines said only 20,000, which is very, very low. But other guidelines are usually like about 5,000 or 50,000 or 60,000 for the platelet count. Okay, but for the math, it's just said 2 minutes bleeding. Okay, and if it bleeds, we have a topical agent, the tranexamic acid mouthwash, so that to help stop the bleeding as well. Right? So why do we have to brush so much? Okay, first, you see that they recommended honey already. So honey can cause dental caries as well, right? And also during the course of chemotherapy, patient may have altered taste, and some people recommend if patient Google for a natural remedy, they would find a lot of recommendation online that you should have the pineapple because they have the bromelain enzyme to break down the protein. They help to reduce the metallic taste in your mouth. So this is also cariogenic, right? If they I have some of my patients use the pineapple juice and make it an ice pop, and they suck on the pineapple juice ice pop all day. Okay? And you can see the set that a friend give to another friend during the chemotherapy. Okay, we're happy to see oral moisturizers and saliva substitute, right? But you can also see a lot of chocolates. You can see Twix, you can see dark chocolates, pomegranate chocolate, because chocolate is also an anti-depression. So it's very common for people to have chocolate during the course of chemotherapy as well. So that's cariogenic as well. So we have to make sure that patients have proper oral care during the cancer treatment. Okay. So speaking of oral balance, management of dry mouth, the ISO defines not just hyposalivation and distinguish hyposalivation and xerostomia, but they also distinguish saliva gland hypofunction as a further step. So salivary gland hypofunction and hyposalivation differs in the salivary flow rates. Okay. And in the latest recommendation, the evidence-based guideline, they still recommend topical mucosal lubricant for saliva substitute, still recommend the masticatory of the saliva reflex, whether it's a sugar-free, low-salt, or any stimulant like acidic non-erosive mouthwash, or oral pilocarpine, still recommended, acupuncture, electrostimulation, like acupuncture, electrostimulation, still recommended. But they do not recommend the extract of ginger or mesenchymal stem cell therapy. Okay. They do not recommend some saliva stimulation that's too erosive or too acidic. Okay, so on the right hand side is okay. There are saliva substitute, but on the left hand side it's saliva stimulant. So you have to be quite careful for how long you use the saliva stimulant, which is acidic. Okay, so I'm going just to show you varieties of the products. Okay. In Japan, they also have a dry mouth clinic. In Thailand, there are some big hospitals that also have dry mouth clinic, but the availability of the products that we have in Thailand is so limited comparing to in Europe or in Japan. So in Thailand, we have Glandosan, which is a bit acidic. We have Biotine and we have GC dry mouth. And also we have oral moisturizing jelly, which is locally made. We also have Cafosol, but it's very, very expensive. like it's written in Thai here that about like 6,000 baht per week. Okay, so what about after cancer treatment? Okay, the adverse effect of cancer treatment also increase the risk of dental caries. Right? So we have to be very careful with the prevention and management of caries after cancer treatment. Fluoride tray has been recommended for a long time with the use of neutral pH sodium fluoride gel. But you can see here that people who have like very dry mouth, some of them cannot tolerate the fluoride tray. So they have to brush the fluoride gel daily instead of using the tray, okay? And the rinse is not enough, is not adequate to have the protection properties for the dental caries in this high-risk patient, okay? And if the mouth is so dry, you don't really have calcium and phosphate, which is the saliva is a reservoir for calcium and phosphate. So you may need to add the calcium phosphate, like topical agent as well, not just only the fluoride. Okay. This is one of my patient who had like part of the tongue removed, and he could not tolerate the fluoride tray, so he had to use the brushing technique instead. Okay. And for the high risk dental caries, we need a higher concentration of fluoride toothpaste. It is recommended to use 5,000 ppm fluoride toothpaste if you are over 16. If you are over 10, you use 2,800 ppm. But in Thailand, we don't have either of this, so we have a locally made 5,000 ppm fluoride gel. Okay. In the States, in Canada, they have the Clean Flow and Colgate Prevalence 5,000 ppm as well. And this product, they have like done or low sodium lauryl sulfate. So there's no foaming, there's no irritant. So the patient can just like do a very minimal post-brushing rinse. And because of the high concentration, we don't have to use a lot of amount of the toothpaste. Okay? And it actually, even though the cancer patient has swallowing difficulties, you don't have to worry about ingestion of the toothpaste much because of the amount that we use is smaller than just regular toothpaste. Okay. And what else? Recall more often. You have to recall the patient maybe every 3 months. Okay. And this is the last part. After the cancer treatment, like the former speaker mentioned, the rehabilitation, whether it's like correcting the scar, the fiber scar, or making the denture, not just for articulation, occlusion, or mastication, but also for swallowing as well. So palatal augmentation processes should be encouraged as well. Okay. Or palatal lift in combination with palatal augmentation processes, okay, for swallowing or also for speech. Okay, so the take-home message is team up. The combination of who are the member of the team depends on the resources of your hospital, and try to get the communication going, whether it's a face-to-face case conference or whether it's a teleconsultation. Okay, start supportive care as early as possible and use the evidence-informed practice throughout the whole experience of cancer care. I don't have time to talk about palliative care of the patient who had no cancer treatment, but there are issues of pain, bleeding, and also the smell of the tumor itself. We can use like metronidazole gel to reduce the smell, and also we can use like opioid like all the morphine and ladder of pain management for the pain. So thank you. And the MAS and ISOO is going to have the annual meeting in Toronto next year. So if you're interested, it's a meeting that's worth going.
Thank you, Professor Mrna, for a very informative seminar. It's an honor to work with you because you not only welcome geriatric patients, you also welcome special need care patients and even post-cancer surgery patients. My question to you is, could you share with us and our colleagues in TUMS, with the rising old age population, how can we incorporate the education about undergrad in geriatric patients, especially about their psychosocial behavior and problems and oral health issues?
So in Thailand, we are fortunate that we have a national policy of incorporating geriatric dentistry within the undergrad level as well. So every dental school is now, including dental nurse school, are mandated to have the course in gerodontology or geriatric dentistry. Like for our school, we have 2 credits of geriatric dentistry and special needs patient as well. In other school, usually it's about 1 to 3 credits, and including the social science side, the psychological side, also biomedical side as well.
Thank you. Would anyone from the audience would like to ask any questions, please?
Just regarding your the last comment by you and by Dr. Mrna, I want to say that in our curriculum also we have a specific course, gerodontology, not gerodontology, but it is for geriatric, not just dental or oral health care of geriatrics. So it is geriatrics course in general, but we have some content in it focusing on gerodontology. And we also have in our, for example, prosthodontics course, for example, in periodontology course, we also focus on specific aspects of gerodontology. So I would like to thank you for your nice presentation.
Thank you. And Dr. Priyapong.
Thank you, Dr. Mrna, to give us very excellent presentation and many things I didn't know before I listened to the lecture. It's unfortunate that I finished dental school too early, so we didn't have any training in geriatric dentistry at that time. So I remember that I came across the patients who had the fluoride tray, and she came to visit dentist because she running out of fluoride gel. And at that time, we didn't have, I think it's very difficult to find a neutral fluoride gel at that time. So I suggest her to use a fluoride toothpaste, but I'm not sure which type of toothpaste that I should be recommend or I should be avoid for suggesting for the patient.
So for the cancer patient, non-abrasive with no irritant, like no SLS, non-foaming would be good for swallowing difficulties, and also high fluoride concentration. But unfortunately, in Thailand right now, Colgate has stopped selling the Prevalence. So now we have the locally made from Cancer Institute, like Maharaj Hospital and Songkhanakarin University that we bought from them. So it's 5,000 ppm gel. You can use it as a tray placement, or you can also use it as a toothbrushing agent as well. And the only brand imported neutral fluoride gel has been suspended because of COVID-19 about like a few months ago.
Okay. Would anyone like to ask any question to Professor Mrna? No. So Dr. Mohammed Kami, I think we can introduce the last speaker for today.
Yes, okay, thank you very much. Last but not the least, of course, I am happy to introduce Dr. Mrna Processing, our last speaker in this event. She is an assistant professor in oral and maxillofacial pathology department. She is also vice head for educational affairs of the department, and her field of activity and interest is odontogenic lesions and also oral cancers. So, dear Dr. Processing, we are ready to hear your presentation.
Hello everyone. Thank you, Professor Khami, for introducing me. Good afternoon, everyone. Hello. You hear me from Tehran University of Medical Sciences. I am Mrna Processing from Oral and Maxillofacial Pathology Department of School of Dentistry from TUMS. Allow me to extend warm welcome to all of you. Welcome to my presentation. It's really is a great opportunity for me to talk to all of you in this valuable webinar. The purpose of my presentation and today I'm here to talk about the microRNA's profile related to prognosis in oral squamous cell carcinoma. The purpose of my today's presentation is explaining some general information about the microRNA's profile, especially in prognosis and diagnosis of oral squamous cell carcinoma. As you all aware about the cancer, today cancer is a big dilemma for all of us. One of every 6 deaths is caused by cancer. Head and neck squamous cell carcinoma stand as a 6th leading cancer by incidence and 8th by death worldwide. And also oral squamous cell carcinoma counts nearly 95% of all head and neck cancer. And unfortunately, as you know, in more than 48% of these patients, unfortunately, being diagnosed in later stages, stage of 3 and 4. And unfortunately, the 5-year survival rate of the patient with head and neck squamous cell carcinoma, especially oral squamous cell carcinoma, is 40 to up to 50%. And about the microRNA, as you all are aware, a big family of superregulatory, small, about 18 to 25, in average 22 nucleotide non-coding RNAs, microRNAs, have regulatory rules in disease. We are regulating expression of protein coding target genes at both post-transcriptional and translational levels. They have high specificity in expression with regards to tissue type and also in tumor characteristics. microRNA's expression profile have better accuracy in disease classification than messenger or mRNA expression profile. As you know about the microRNAs, abnormal expression of the microRNAs detected in tumor, in serum, in plasma, or in a saliva sample from the patient with oral cancer has clinical significance in prognosis, prediction, and also in development of the effective treatment. Because microRNAs are involved in a variety of cellular processes, as you see in this picture, including the regulation of cellular differentiation, proliferation, apoptosis, angiogenesis, and resistance to cell death or activation or initiation of the process of metastasis in a cancerous cells. Most of the present literature on oral cancer specific RNAs comes from European and American study subjects. And as you know, I'm sure that you confirm me that microRNA expression profile studies demonstrate difference among population. Despite the growth in literature on microRNA and their specific expression status across tissue and also disease types, each new study bring up a new profile with a very few common signatures. Ethnic variation in the study subject have been found to be a major contributor to this difference. And beside a few tissue-level microRNA expression study, majority of the data is derived from oral cancer cell lines. Inadequate in provide a significant picture on microRNA expression status. And I'm sure that everybody has heard about the inconsistent microRNAs profile in the literature. I think that the most important reason for this situation is the type sample, including frozen tissue, formalin-fixed paraffin embedded blocks or cell lines, and localization and origin of the lesion, oral cavity, and other anatomical site in the head and neck, and platform used, and also lesion type analysis from a premalignant lesion to different histopathologic grades of oral squamous cell carcinoma to advanced lesion with metastasis. And you can see here about the classification of the microRNAs in cancer. We can classify microRNA in two big group of tumor suppressor microRNAs and also two oncogenic microRNAs. And we can use with this classification rule and application of microRNAs. In addition, the aberrant expression of the microRNA is known to induce various human cancer and human malignancies clearly classified by their microRNA profiles. And for example, specific overexpression or underexpression of microRNAs have been correlated with particular type of tumors. It has been suggested that microRNAs overexpression could result in a downregulation of tumor suppressor genes, while underexpression may lead to oncogenic upregulation. But however, in this situation, little is known about the microRNA expression pattern or also functions in oral cancers. Most importantly, it has been suggested that microRNAs expression signatures could be used to predict the outcome in several tumor types, including lung cancer and also CLL or chronic lymphocytic leukemia, as well as to predict the response to chemotherapy. In the ocean of the researchers in the field of microRNAs in oral cancer, I selected some of them with the most citation in literature, and also I presented two of the our research in our department about the microRNAs in field of oral cancer. The first one is the a paper by Soga et al. At the, I think, 2013. Yes, in 2013, Soga et al. Reported that 12 microRNAs, the list that you see here, such as microRNA number 31 or 21 and the else, were significantly upregulated in most of cancer tissues samples compared with the normal oral mucosa. And on the other hand, 11 microRNAs, such as microRNA number 133A and the else, showed downregulation in cancer tissue sample compared with the normal oral mucosa. You can see here the table that Soga reported in his paper, that you can see a list of microRNA that's by up or down regulation in oral squamous cell carcinoma. Significant value by p-value analysis that associated with pathway or target genes in head and neck, or especially oral squamous cell carcinoma. For example, you can see here microRNA 29A with downregulation we can example microRNA number 181A, which downregulation of this microRNA associated with KRAS pathway and also with twist gene in oral squamous cell carcinoma. And here in our center, I would like to present two of our research in our department, oral and maxillofacial department of TUMS in field of oral cancer, and especially evaluation of microRNAs in oral cancer. The first one that you see here, Dr. Bahri and Dr. Shakib et al. Reported that microRNA 26B might have a tumor suppressor role in oral squamous cell carcinoma. And they reported that this effect may be a result of inactivation of PTEN, well-known tumor suppressor gene. They reported that this microRNA and PTEN can be used as a prognostic biomarkers in initiation and also progression of oral squamous cell carcinoma. And in the other researches that I presented today, Karmi et al. Evaluated 3 types of microRNAs in oral squamous cell carcinoma by serum samples from these 3 types of microRNA, circulating serum type of microRNA number 21 and 24 and also 29A were evaluated in this study, and our results showed that microRNA number 21 and 24 were significantly higher, and microRNA number 29A was significantly lower in plasma of the patient with oral squamous cell carcinoma in comparison with the normal group. And in the last study I presented here, Rajan et al. In the 2021 reported in a large evaluation of microRNAs study, a set of 105 microRNAs to be differentially expressed in oral squamous cell carcinoma. He reported that out of which a subset of 19 most dysregulated microRNAs that were validated by quantitative real-time PCR. He reported that upregulation of some microRNAs number 196A and number 20A and also 1237, and downregulation of some other microRNA associated with poor prognosis of oral squamous cell carcinoma patients. And in addition, he reported that upregulation of some upregulation of some upregulation of some microRNA number 196A and downregulation of microRNA number 204 as a best predictor for disease recurrence and also for patient survival. You can see here the table which the Rajan et al. Reported in his paper and a list of microRNAs that he evaluated in his research. That you can see here microRNA number 1237 that among the other microRNAs, this tumor specific overexpression of this microRNA was a novel report, while the other signature microRNA like microRNA number 21, like our research in our department, and the other have been studied and reported in human cancers, including oral squamous cell carcinoma. But this microRNA was a novel report until now. And also, you can see here that both microRNA number 196A and B have been proposed as a biomarker in cancer management, and their expression was found to be prognostic in cancer cell migration and invasion and also lymph node metastasis. And in Kaplan-Meier survival analysis, reveals that his study reveals that the association of microRNA number 196A and also microRNA number 204 individually with overall and also disease-free survival. About the microRNA as prognostic biomarkers, in ocean of the research, there is several number of researchers and reported that I'm sure that you confirm me about in this project that the expression pattern of certain microRNA have been RNAs have been found to correlate with clinical stage, lymph node metastasis, and also patient survival, indicating that these microRNAs can act as a prognostic predictor in oral squamous cell carcinoma. For example, you can see here an example of this rule for microRNAs, that higher expression level of microRNA number 21 in tongue squamous cell carcinoma correlated with advanced clinical stage and also with poor differentiation and also with lymph node metastasis. However, in this field, a multivariate analysis shows that expression level of this microRNA could be used as an independent prognostic factor for tongue oral squamous cell carcinoma and associated with survival of the patient. You can see here a list of other microRNAs like microRNA number 31 and also 125B and the others that these microRNAs were found to be associated with lymph node metastasis and poor prognosis and poor oral squamous cell carcinoma patient survival in the literature. In the other hand, in the field of microRNA as a target for oral squamous cell carcinoma therapy, you can, as you all aware about this, this fact that the ability to manipulate microRNA's expression and also functions by local and systemic delivery of the microRNA inhibitor like anti-microRNA oligonucleotides or microRNAs sponge has recently gained interest as a novel therapeutic approach. Maybe you know about the history of this treatment. The treatment approach came into light after the first successful treatment approach about the microRNA, anti-macro RNA oligonucleotide-based human clinical trial in 2011, I think, for the treatment of hepatitis C virus infection. After that, recent identification of K microRNAs and either oncogenic or tumor suppressor functions in oral squamous cell carcinoma has opened up a new possibilities for microRNA-based oral squamous cell carcinoma therapy. And you know that the advantage or the major advantage of microRNA-based cancer therapy lies in the ability of the microRNAs to target multiple forms of pathways involved in cell proliferation, differentiation, angiogenesis, or survival of the pathway involved in the cancer cells. The example for this treatment therapy, I read in the literature two microRNA number 21 and also microRNA number 99A, which inhibition of this microRNA can support it and suppress the proliferation and also induce the apoptosis in the tongue squamous cell carcinoma cells in vitro studies. About the resistance to chemotherapy and radiotherapy, the resistance to chemotherapy and radiotherapy is one of the major challenges in the management of the oral squamous cell carcinoma, as significant high proportion of the oral squamous cell carcinoma lesion fail to respond to this treatment modality. Recent study have linked this resistance to chemotherapy and radiotherapy in oral squamous cell carcinoma patient to altered microRNA expression and also function. You can see here the list of downregulation and also upregulation of some microRNA reported in the literature that in head and neck squamous cell carcinoma cells, these microRNAs have correlated with multiple drug resistance and also in phenotype in vitro studies. In addition, microRNA signature or signatures related to therapeutic resistance have also been used experimentally to reverse the resistance phenotype. For example, as you see here, inhibition of microRNA number 21 by anti-microRNA oligonucleotide has been shown to inhibit chemoresistance in oral squamous cell carcinoma cells. And also, likewise, forced expression of some microRNA, for example, microRNA number 125B, has been reported to enhance the radiosensitivity in oral squamous cell carcinoma cells in vitro studies. And there is an important point in here about the treatment and about the in field of resistance to chemotherapy and radiotherapy in oral squamous cell carcinoma, that before the conclusion, I would like to explain this fact that today, recently, nanoparticle based delivery of microRNAs was suggested as a promising approach in treatment of head and neck and especially oral squamous cell carcinoma. Despite this promising result, more studies it's very clearly that more studies are necessary to better understand about the effective delivery system to optimal uptake and also to decrease or to minimize degradation of the microRNA-based drug in the in vivo situation, as it's very clear that in vivo situation is very different from in vitro situation, and maybe the challenges was very big sometimes. And finally, in conclusion, you can confirm me that alteration in the expression pattern of microRNA is a common finding in oral squamous cell carcinoma, and it's a big challenge ahead to translate this promising finding to clinical before the following issues will be fully addressed. And maybe specific microRNA signature identified from the tumor specimen or from the serum specimen or plasma specimen, or also saliva specimen from oral squamous cell carcinoma patient have a potential to be clinically useful in the diagnosis and prognosis and therapeutic targets in oral squamous cell carcinoma. And finally, thank you for your time and your attention. I really wish my presentation be informative and also interesting for all of you. Thank you very much, and God bless you.
Thank you, Dr. Darakhshan, for your nice presentation and knowledge and information you shared with us. I'm so happy that we are on time after 8 lectures, and this is fantastic. So we have time now for question and answer. If there is any question, please just raise your hands or open your microphone and ask your question. Dr. Darakhshan, if you just stop sharing your screen, then we can have a better view of the participants. Yes, thank you. Okay, so okay, so
hello. May I ask a question?
Hello, sure.
Thank you, Dr. Darakhshan, for your great presentation, focusing on microRNAs. Would you please, unfortunately, I lost few minutes of your presentation, and would you let me know if there is any anti-microRNA drugs, and is there any drugs developed to target microRNAs, especially in oral squamous cell carcinoma?
Yes, of course. Thank you, Dr. Shahid, for your attention and for your time. Yes, of course. About the anti-microRNAs in treatment for targeting the oral squamous cell carcinoma in therapy, we can focus on 2 studies about this microRNA, that microRNA number 21 and also microRNA number 99A has been reported and published in literature in treatment for tongue squamous cell carcinoma in vitro studies. And I opened here the paper, for example, the inhibiting the microRNA number 21 by anti-microRNA oligonucleotides inhibits the survival and anchorage dependent growth, and also inhibits and decrease the invasion in oral squamous cell carcinoma cells. And in the other hand, as I mentioned, restoration of microRNA number 99A level by microRNA mimic transfections markedly suppressed proliferation and also invasion, and also induce apoptosis, means increased apoptosis in tongue squamous cell carcinoma cells in literature. I find these 2 recently published papers about this new field of therapy in oral squamous cell carcinoma. I think it's a new big windows to treatment of oral cancers.
Okay, thank you very much. Also, I would like to let our colleagues from Thammasat University, that faculty members at oral and maxillofacial pathology department are pioneer in our country to conduct research, oral squamous cell carcinoma associated researches. Therefore, on behalf of our colleagues in oral and maxillofacial pathology department, welcome any collaboration and joint research on this topic. And I would like to ask also Dr. Derakhshan to be in touch with respected colleagues and professors from Thammasat University to make and conduct a joint research on oral squamous cell carcinoma, focusing on pathologic features and also microRNAs associated researches. Thank you, Dr. Khami.
You're welcome. Thank you. So is there any other question? Okay, so yes, Professor Vipavi.
Hello, thank you very much for your excellent presentation. I just wonder if there is any microRNA that we can use as a biomarker for early detection of oral cancer.
Thank you so much for your nice question. As we know about the detection of the cancer, the literature is very little about the detection of the oral squamous cell carcinoma, but by microRNA expression. Several number of microRNA overexpression or underexpression has been detected in oral squamous cell carcinoma, but any of them is directly guide us or lead us to the detection oral carcinoma in the earliest stage, or any of them until now is now a suitable way to detect the oral squamous cell carcinoma in vivo studies. But very often is downregulation or upregulation in oral squamous cell carcinoma. But for detection, no. Based on my review of literature, any of them is used now in the in vivo or in clinical setting for detection of oral squamous cell carcinoma.
Thank you.
Hello.
Hello,
good to have you, Dr. Derakhshan. Please, I would like to ask about this microRNA treatment in oral cancers. Is it possible to apply the same treatment modalities in other oral cancers, such as osteosarcomas, rhabdomyosarcomas, and other forms of sarcomas, please? Thank you.
Thank you so much for your question, Mr. Bashar. I don't read any paper about the microRNA expression or aberrant expression or dysregulation of microRNA in other sarcomas. It's very clear that in a malignancy situation, like sarcoma, or it's not different in sarcomas, also osteosarcomas or rhabdomyosarcomas, you mentioned that really there is a number of dysregulation of microRNAs in all the malignancy situations, like sarcomas, like carcinomas. But I don't read recently about the dysregulation of microRNAs in osteosarcoma or rhabdomyosarcoma. I think it's very necessary to read the literature, and also I think in this field of sarcoma, based on the difference in treatment of these lesions with carcinoma, it's very important to increasing the knowledge about the microRNAs, because these sarcomas respond very better to chemotherapy, and also with drugs more than more than carcinomas. And in carcinomas, it is a big challenges to respond to chemotherapy and also radiotherapy. But in sarcomas, we have less challenges about this approach treatment. But I don't know. And now here, based on the review of literature in recently day about the sarcomas in field of microRNAs.
Thank you so much.
Thank you, Fran.
Okay, thank you very much. Please accept my apology. I was disconnected and then I came back. So is there any other question? Okay, it seems that there's no more questions. So, okay, now it's time for concluding and closing remarks of this event. So I would like to ask Dr. Beharat to start this part of the event.
Thank you, Dr. Mohammad Kami. I would like to thank everyone who participated. I would like to thank all the speakers. And I would like to thank personally Dr. Piapong, who gave me this opportunity. And of course, the Dean of both of our universities. And I hope we have more such collaborations together. So,
okay.
Yes, Professor Siriwan, please add your comments.
Mm-hmm. Yeah, I just would like to have a very quick comment. Since I am Vice President for Research and Innovation of Thammasat University, and I would like to propose, the possibility that we can initiate the joint research project between our universities. For example, at Thammasat University, we have the grant, which is the matching fund. Matching fund means, for example, Thammasat will fund our researcher, and your university also fund your researcher, and then both of them can conduct the research in the same direction. And of course, both of them publish papers together. So we can start our collaborative project by this matching funding that we can provide from our university. So that is one possibility that we can do. Thank you.
Thank you. Thank you very much for your great suggestion. I think it's a great opportunity for us to have collaboration with Thammasat University. And I think it's possible to follow this suggestion and try to establish some joint projects. Thank you very much.
May I add a little comment after we finish the webinar? I think this webinar is very insightful webinar, and it expands our knowledge and vision from, I would say, cellular level to clinical level and also community level. And it is our first joint webinar between Tehran University of Medical Science and Thammasat University. And clearly it is a successful that will lead to other joint conference and also collaboration. We are lucky that we have Dr. Siriwan with us. So Dr. Siriwan is in charge of all research funding of Thammasat University. And for the proposed project regarding the pathological research in microRNA, I think we have a matching specialist like Dr. Samira and Dr. Ekarat. So it is very high possibility. And also with Dr. Vipa, we that we can develop some proposal together, and also artificial intelligence and clinical detection of oral cancer with Dr. Grisasit, and also about the oral health support and oral health care policy with Dr. Mrna. I think that is some area that we can further develop for future collaboration. Thank you.
Thank you, Doctor.
Please continue, Dr. Bharat.
I would actually also like to add that I would be interested if Dr. Warin could have some joint collaboration or a workshop about introducing us his AI, his research about the AI more. We have.
Would you let me add a comment? I think it is not the end of the webinar, but it is the beginning of our upcoming collaboration, and it was a very informative for me, and I think we could make a virtual bridge between two universities, and we definitely follow this bridge. And also I would like to express my sincere appreciation to you all, especially deans of both schools, Professor Inglam, and also my very enthusiastic colleagues, Dr. Piafong and also Dr. Baharat. Also, I would like to express my sincere appreciation to Dr. Mohammed. He always kindly supported us at the International Affairs Office, and thank you all.
Thank you, Dr. Shaki. I would like our Dean, Professor Inglam, to speak a few words. Professor Inglam, would you like to say something? Oh, I think we. I think there is some technical problem at the dean side. Actually, we also have among us the specialist in maxillofacial prosthodontist, Dr. Ray. Dr. Ray, you heard any comments from you?
Hello?
Yes, we can
hear me. Yes, hi. Thank you for all the great presentations today. Can you hear me? Thank you for all the great presentations today. I happen to be at the hospital working clinically as well too. So, at Bumrungrad International Hospital here in Bangkok, we are doing some clinical research as well too. So if you have any collaboration opportunities on the clinical side, the hospital would be quite interested as well. So thank you all very much for the great presentations today. It's very interesting.
Thank you, Dr. Ray, for participating in this session.
Thank you.
Okay, so I would like also thank all the planners of this fantastic event from both sides. Dr. Piafong, Dr. Bharat, and Professor Inglam, who kindly supported this event, and also all those who were engaged in planning and holding this webinar. In our side, Puyan and his team, his technical team, I think all the team from both sides acted great, and the result was the event that we had. And as Dr. Piafong said, we had journeys from cell to society to clinic, back and forth. And this is the way that we should control the diseases at community level, I think. I'm sure, as you all mentioned, this is not the only step of collaboration, but maybe the first step, and I'm sure that we can continue more fruitful collaborations in the future. I hope that we can one day, with improvement in the situation of the pandemic, we can one day have in-person maybe seminars in Tehran or Bangkok or in both, I hope. So, and now I just want to say that we have the certificates prepared, and I ask my colleague, Ms. Sadeghi, who she is always supporting us in these events, and I want to thank her also being with us during the whole event, just to share the certificates one by one. And this is just a primary appreciation of all the lecturers who kindly assigned their time and efforts to prepare great lectures and present their lectures in this meeting. So first, Dr. Ikarat. So please give your hands for him. Maybe we can, yes, okay. So thank you for being with us and for your nice lecture. Then Dr. Katayun Sargeran, again. And our next speaker was Dr. Warin, Professor Warin. I'm not good at spelling the name, but the family name is easier for me. So Dr. Warin, thank you for being with us with 2 lectures, very informative and good lectures. Thank you. And Dr. Sara Pourshahidi for her nice presentation on laser therapy of oral cancers. So, and then we had our colleagues here, Dr. Mrna regarding reconstruction of oral cancer lesions. And then we had Dr. Matana for rehabilitation or care before, during, and after cancers. Very informative, very good lecture. And then we had Dr. Samira Derakhshan, who presented some new insights regarding microRNA. So that is all. Thank you all for being with us. We are ready if there is any other, okay, yes, give your hands at the final. Yes, okay, thank you. And I also want to thank Dr. Baharat for coordinating and for moderating this event from Thammasat University. And yes.
And this is yours, and this is yours. The next one is yours, Dr. Mohammed. Thank you for helping us and supporting.
Okay, thank you. Thank you very much. Okay, so we are at the end of the event. So if there is any final comments by any of the participants, we are more than happy to hear. Okay. So if there is no comment, please just turn on your cameras to have a group photo.
Yes, please, everyone, please turn on your cam to have a group photo at the end.
Okay, thank you all.
Okay.
Miss Sadeghi, would you let's see the video of all participants.
Okay, very good.
Okay, thank you. Great. Thank you.
Okay, thank you all for your participation in this meeting, and I would like to say goodbye to you.
Thank you. Thank
all
you.
of you.
Have a nice day. Thank you for your contribution.
Thank you.
Thank you.
Bye.
Have a nice day.
Nice day.